Good evening. Welcome to Hampton Bays High School's 11th Annual Science Research Symposium. My name is Lars Clemenson. I'm the Superintendent of Schools of this fantastic school district, and we are super glad to have you as our guests tonight. As we begin, I'd like to take a moment on behalf of the Board of Education and the entire school district to wish each of you well and welcome you here. In so many important ways, the Hampton Bays Schools is committed to continuing to move forward with applied science. Tonight, you'll experience the same sense of optimism that we in the Hampton Bays Schools have for the future after you meet our science researchers. These students will play a very important role in the future as our nation and our world tackle very real challenges that we face today. We're taught in school that the core of science, and science research specifically, is to question, to consider, and to seek truth that's based in data, research, and the scientific process. Now in its 11th year, science research has served many students on this journey, but has had an institutional impact on the Hampton Bays School District. We've introduced the Advanced Placement Research and Seminar Series as part of our capstone program, and also our News Literacy Initiative. We teach students to question and verify on a daily basis. And as the years go on, Hampton Bays High School is committed to personifying the word applied in all aspects of what we do, making life and learning a practical, project-based experience. Tonight, you'll meet students all along the way in their science research journey. Like the yellow brick road that leads to Oz, each brick builds towards a more precise and more fulfilled question that our students have become passionate about over their 4 years. Among our researchers tonight, you'll meet kids who are still toying with their topic, pawing at it to see if that's a particular angle that they're inspired by, that they want to learn more about. And you'll meet scientists and researchers ready to graduate with a very clear vision for the science question they want to tackle, and some who will tackle it in college. They're already engaged in the university and the field with a mentor, and they're starting to accelerate and fine-tune that research from 9th grade all the way to 12th grade and beyond. Every step of the way in science research is critically important and challenging. From learning how to read peer-reviewed articles, from learning how to find peer-reviewed articles as a 9th grader, to crystallizing what students learn, to be able to come up here tonight and present it to you, is a tremendous lift over 4 years. The science research program is the most challenging program and course of study that a student can take in the Hampton Bays Schools. It's done over 4 years in small groups, with a mentor, and with a tremendous amount of independent study. Under the very best conditions, this is rigorous and challenging over 4 years. I'd like to acknowledge some special guests tonight who are both watching with us and present with us tonight. Representing our Board of Education, I'd like to welcome Trustee Ann Colhane. And leading across the canal at Southampton Town Hall, welcome to Councilman Michael Iacilli. Thank you for being here. I'd like to recognize both Mr. Richard and Mr. Morgan, our building leaders, for their support of science research. And I'd like to offer a special acknowledgement to our mentors in the field, who are both local and from around the nation, who have willingly committed to our student researchers. There's no obligation to these mentors to extend themselves, their time, their resources, in some cases their graduate students, and their laboratories to our students, yet they do it for the love of their research and the love of feeding the pipeline for future researchers. These mentor relationships are critical to paving the way for our students' future. I'd like to recognize Senior Dylan Foley's mentor tonight, Matt Sclafani, a marine educator and researcher from Stony Brook University, and thank him for being here tonight, representing our mentors. Thank you. Finally, I'd like to credit the university and the high school program at the University of Albany and its directors who are watching from afar. We value this tremendous partnership, and we appreciate the commitment to Hampton Bays. Our science researchers will graduate from Hampton Bays High School with 12 college credits from the University at Albany. Remarkable start to a college education. I want to thank Ms. Mary Beth Lavazzoli for her coordination of this program, all of the members of the science department from kindergarten through 12th grade, including our newest partner, Dr. Dmitry Zaslavsky. A very special thank you to our science department coordinator, Ms. Christina Fassini. It was Ms. Fassini's leadership and the tremendous work that we've both seen and haven't seen that's been behind the curtain and in evenings and weekends that have brought tonight's event to life. Ms. Fassini, thank you for getting us to the stage tonight. Our students are ready to introduce you to their research questions and share their passion with you. You will leave tonight, if we do our job well, with more questions than answers, but thankfully more certain and optimistic and proud, number one, to be from Hampton Bays, but number two, for what our future holds. Thank you all for being with us tonight. Enjoy your time here. You are always welcome, and we always treasure the opportunity for our community to engage with our students. I'd like to now introduce our keynote speaker tonight, Chris Paparo, Stony Brook, Southampton's Marine Science Center Manager in the School of Marine and Atmospheric Sciences. Just a few words about Chris. His primary responsibility is to maintain the Marine Science Lab in Southampton. Their computerized, state-of-the-art 5,000 square foot seawater lab is real applied science. He's also the field station's naturalist, educating the public and community about the various fish, invertebrates, mammals, and birds that call Long Island home. Notably, Chris brings 20 years of experience working and educating in our community. He's tagged white sharks with Osage, and he can share some stories, I'm sure, about our busy shark seasons that we've been experiencing. Ladies and gentlemen, please help me welcome to offer his talk, Wild Long Island, Mr. Chris Paparo.
All right, well, first I want to say thank you for inviting me to come out to speak tonight, but I also want to say how jealous I am of this program you guys have here. In high school, I didn't have this. We had a marine science course that was an elective that you took because you didn't want to do anything hard, you know, and I would have given anything to have a marine science program that you guys are experiencing today, so I hope you really, really, really appreciate it. So. Some of you might also know me as Fish Guy, of Fish Guy Photos. That's my other alias. And one of the things I like to do is, through my social media and through my writing and lecturing, is I like to talk about the positives, the cool things that are happening out there. And I thought I'd continue that tonight, especially for you guys, because you often probably see a lot of times in the news toxic tides and dead turtles and all these things that are negative. And it seems like it's, why bother? You know, there's so much stuff that is never going to get better. What difference am I going to make? So I thought I'd show you just a few things that scientists have made a big difference, and we'll be passing the baton off to you guys to be the future of making things right. So well, the first thing we'll talk about is one of my favorites, and it's here now, and this is definitely a thing for the Hamptons and Hampton Bays. Uh-oh. There we go. The osprey, you know, this is a sign of spring. Summer is almost here when we know the ospreys show up. These are fishhawks, but one of the things is, you know, these are very common. In fact, I have a pair right outside my office, Ral and Mary. Ral's on the top, Mary's his mate there on the bottom. But osprey are super common, but when I was younger, that wasn't the case. Their population was almost obliterated due to the use of DDT, which was a chemical they used to kill mosquitoes. Well, back in the 70s, a bunch of researchers got together and said, you know what, this stuff is bad, it's poisonous, it's killing our environment, and they banned it. And within a short matter of time, we started seeing osprey make a return, and we started seeing baby osprey as well. And one of the things DDT did was it would make their eggshells so thin that when the female would incubate her eggs, she would just crush the chick and kill the chick that way. So again, without DDT in an environment, we're starting to see these osprey make a return. Another critter, I'll just call them critters at this point, but another bird that can thank us for getting rid of DDT is the bald eagle. This is another bird that up until recently, you could not see them. Now, I can find a bald eagle if I go out and look for them, but they're fairly common. You know, you could see them soaring overhead. Most people just don't see them, but they're so common. They're even nesting around here. Scenes like this, again, are very, very common. There's mom with 2 babies in this nest, and this was out here on the South Fork. You know, so again, a lot of work went into this, not only just banning DDT, but you know, they also went to Alaska, and we got eagles to bring back to New York and kickstart them. And again, clean environment, they made a comeback. Some other stuff that we've started to see, fish ladders. You know, we've dammed up so many of our rivers and streams throughout the world, and now we're trying to make it right, and we're trying to fix it. There are many, many fish that need fresh water. They spend their adult life out in the ocean, they come back to spawn. In this case, this is a fish passage that's in Riverhead, and here's a little bit of look at what's going on, and it's happening right now. You can go into Riverhead and see this very thing. So these are alewife, and alewife are a type of herring. They're a sea herring, and a river herring they call them, but they spend their adult life out in the sea, and they come back each spring to fresh water to swim all the way into the fresh water where they'll spawn and then turn around and head back out again. And you can see these fish here are going up current. And in a moment, I'm going to show you what it looks like for their side of it, of heading up this passage. You know, the dam that's there is about a 4-foot dam. There's no way this little fish can jump 4 feet. But by putting a passage in, they'll swim up. And then this will then benefit osprey and eagles and otters and all sorts of other animals that live up in the fresh water. There's the ladder. Now they just have a little tiny bump to go, and they can get up into the fresh water. For years, people would just bucket these fish over, but now, again, by doing the right thing, they're getting up there. These ladders also help in other species, and I just filmed this last week. Those are glass eels. Glass eels are the larval stage of the American eel. They are spawned out in the middle of the Atlantic Ocean, and these little tiny glass eels swim all the way to shore to go into fresh water. And the same problem with the alewife they face is there's dams and blockades. So these ladders now help these glass eels get up into the fresh water where they'll stay for a couple years. They'll then go into the bay for a few more years until they go back to the ocean to spawn. It takes almost 50 years for these, or up to 50 years for this one eel to go back to the ocean and spawn. And again, we're fixing this, and look at all these eels that are there. This is probably the most eels I've ever seen. And again, this is a good sign of healthy habitats and fixing things that were wrong in the past. Seals, they're leaving now. They're one of our winter residents, but when I was a kid, you could not find a seal. You know, and if you did, you were so excited. It was like the highlight of the winter because they were so rare. Now, this is a fairly common scene. Some of you might have been out with me on our research vessels at Stony Brook to look at these seals. You know, again, they were hunted to near extinction. Some species were wiped out from the United States, but again, through protecting these animals and bringing back the habitat, they're now making a return. These ones have been all over the beaches right now. This is a gray seal pup. They're born in the winter, and after 3 weeks, they're on their own. Mom lets them go. They're gone. They have to fend for themselves in the dead of winter. And right now on Dune Road, last week there were 10 that the New York Marine Rescue Center was observing because these little seals get tired and they sleep up on the beach to rest. But look at those teeth. You don't want to get close to them. But again, this is a species that was wiped out from the United States and now is returned, again, due to that work, hard work from scientists. There's a bunch of adults, fully grown. They get 800 pounds. You know, this is off of Little Gull Island off of Orient Point. So again, protections work. Another thing that we've seen a lot of in the last couple of years, bunker. The Atlantic menhaden. It's a type of herring. And again, when I was a kid, we didn't see many of them. They were there. We caught them, but nothing like this. This was a school that I filmed right outside of Mecox Bay out in Southampton, so a little further east of here. But this is just an enormous school, and these schools go up and down the beach. And this fuels so much more of our local ecosystem. But we're seeing this now because we've cleaned up our oceans. We've regulated how many of these fish are taken from the ocean. We've realized the big picture. And again, because they're here, we're seeing stuff like these dolphins. These are bottlenose dolphin. Again, when I was younger, they weren't super common. But now, again, Marine Mammal Protection Act protected it, made it illegal to hunt and harvest these animals. And through the protections, they are making a comeback. And then we protect their food, or we regulate the harvest of their food, the bunker, and you get to see scenes like this. So that's a school of bunker getting harassed and chased by a pod of bottlenose dolphin. Now I'll tell you, I'd hate to be a bunker. Everything eats you. In this case, watch, they're just smacking them with their tail, stunning them and picking them up and eating them. Those osprey and eagles I talked about earlier, they sink 8 hooks into their back, fly them to a telephone pole, and pluck their eyes out. It's horrible. Bluefish, 1 bite and they're ripped in half. Striped bass, swallow them whole. But again, these fish, even though everybody is chasing them and wants a piece of them, they are the most important fish in our local ocean. And you can't even eat them. And if I see you can't eat something, it really means you can't eat anything, because I eat anything and everything. But we start seeing them because of that stuff. More protections. This is a very common scene around here. I'm sure everybody, or many of you have probably seen a whale locally on the beaches. That's the beach in Southampton right in the background. And these are common because, again, of protections. The humpback whale was also hunted to near extinction, but due to the Marine Mammal Protection Act, cleaning up of our oceans, this animal is no longer listed as endangered or threatened. It's listed as vulnerable because it's a long-lived animal and it could go the wrong way if things are not if it's not watched after. But these kind of scenes are common. And what you see here is it's eating bunker. And they, instead of smacking them with their tail, they just lunge feed, it's called. And they open their mouth and they just scoop them up. But again, look how close to shore. You can sit on the beach and watch this kind of stuff. 2 of them. My best morning, and when I say morning, it's just my morning before work. I'm usually on the water by 5:30. I'm back to the dock at 8:30 so I could be at work. My best morning, I had 12 whales, 12 different humpback whales, just from Shinnecock Inlet. I can get to about Dolphin Lane before I have to turn around and head back to get to work. So I thought I would end with this video compilation of some lunge feeding. So those bunker, again, are why these whales are here. And the whales are here because we protected them through research and studying these animals. We were able to learn about what they need. Again, not only food, but environmentally. And it's a huge, huge success story. And again, it's because of scientists. And you guys are the next generation of scientists. And there's still a lot of problems. We're not out of the woods at all. But I just wanted to show you that there's been a lot of positive and a lot of success. And we got to keep on that momentum and keep moving forward. And I don't know about you guys, but it's my video and I could still watch this for hours and hours. And on this particular one, I end up slowing it down so you can see it in slow-mo. And I zoomed in a little bit more. That's a big belly full of fish, you know, and talk about heartburn. Oof. All those little fish kicking and swimming around in there. But again, this is right here in our own backyard. And one thing that I'll also mention too is when I was younger and as a student, we were often taught that we have to protect the rainforest. We have to protect the Great Barrier Reef. We have to protect, the plains animals in Africa. And I agree, all of those ecosystems need protecting. But you know what? It's hard to protect something when you're so far away from it. I think, and I wish we would do this, and there's a bigger push for this now, is to protect our own backyard. If everybody protected their own backyard, the jungles, the Amazon would be fine. Africa would be fine. The Great Barrier Reef would be fine. So again, it's worth protecting our backyard. It's a great place. It's full of amazing wildlife. I could speak for hours, I usually do, about the wildlife that's found here on Long Island. But again, if we focus on our home and everybody did that, I think the world would be a much, much better place. So again, just a quick plug for me. There you go, Fish Guy on social media. If you want to learn more about the cool animals that live on the land, air, and sea of Long Island. So congratulations to you all. This is great. And I'm super excited to see what you have to present. So thank you.
In Hampton Bays, we believe in what's called the living classroom. So we do that with honoring our veterans and work that we do in the hospitality industry. And there's no truer example of that than the work we get to do with Fish Guy himself. And Chris has taken our students out on the water with our science teachers and our science students. And I know a number of you sit on Ponquag Beach in the summer. We never saw the whale come up from an aerial view, right? It's amazing to think that we sit and we enjoy this beautiful place, and that's happening just a few hundred yards off the shore. That's what the questions our kids ask are all about in the science research program. This isn't just knowledge for fun. This is knowledge for action. And as Chris charges us all, it's to protect, to answer a question, to advance an understanding or a technology. The beauty of the science research program is that it's student-run. And at this time, I'm going to turn over the microphone and sit back and relax with you as we learn and enjoy from our students. I'd like to introduce you to Samuel Blackburn, a science research freshman, to kick off tonight's student presentations. Samuel.
Our first presenter of the night will be from Sophomore Vasyl Datsuk, whose research is on advancements in machine learning.
So my name is Vasyl Datsuk. I'm going to talk to you a little bit about machine learning. So I want to give some background on machine learning. Machine learning is a computer algorithm that analyzes a set of data for patterns and sort and compute data that have common features. Machine learning is a subsection of artificial intelligence and is arguably considered one of the most important. Machine learning algorithms require thousands of data sets for training, which will be guided on feature and pattern recognition. Primarily, my research this year has been focused on data collection and some possible limitations it could propose to my future research in this area. Due to the complexity of this field of research, it is only possible with some outside guidance. I'm applying to several machine learning experts, such as Angel Bagel. Angel Bagel has graduated from the University of Berkeley with a PhD in computer science. He's also an associate professor of computer science in the Software and Societal Systems Department in the School of Computer Science at Carnegie Mellon University in Pittsburgh, Pennsylvania. Bagel conducted a study for Microsoft in which he aimed to find some possible areas for improvement with data collection and some possible future areas of research. Bagel surveyed 14 senior Microsoft engineers with targeted questions about how machine learning is used in the field, issues with it, and if they could provide a feasible solution to proposed issues. This table here shows the roles of 14 different machine learning researchers. Each participant has varied questions depending on the field of work in Microsoft using machine learning and whether or not they were a field manager. In the study, the most commonly received limitation was the need for more data reusability throughout the different fields. Most researchers suggested that reusable data would seriously benefit Microsoft and machine learning implementation. If data could be used across multiple fields in Microsoft, the initial research from the study shows that it could improve accuracy and decrease costs of using machine learning. In the study, the researchers came to a consensus that improvement in accessibility, accurateness, and definitiveness would lead to a general increase in efficiency and possible field expansion of machine learning. It was also proposed that in the future, machine learning algorithms could be used to create data sets to train other machine learning algorithms. In the future, I hope to use machine learning to detect cancer and other diseases within the human body, and I hope to use this research that I've collected this year to help understand my topic further. Thank you.
The next presenter is sophomore Cooper Foley. He is presenting his research on sleep apnea.
Hello, everybody. My name is Cooper Foley, and I will be presenting on obstructive sleep apnea. My current research topic is sleeping disorders, which this year is focused specifically on obstructive sleep apnea, or OSA, which is characterized by the obstruction of the upper airway during sleep as a result of some form of blockage, preventing air from moving through a windpipe. My research has led to me to look into various different subtopics, such as hypoglossal nerve stimulation, uncontrolled blood pressure, and Down syndrome. A potential mentor to help further my research is Maria V. Sarna, who is an otolaryngology, or an ear, nose, and throat specialist working at Cornell Surgery. I hope to speak with her to narrow down my current research into focusing specifically down onto hypoglossal nerve stimulation and its effects on OSA. An article that I've read by her is The Outcomes of Hypoglossal Nerve Stimulation Therapy, the Current Practice, Future Directions, and Research Gaps. The purpose of the discussion included within this article was to evaluate hypoglossal nerve stimulation for OSA treatment. And the article summarizes various presentations and discussions from the 2019 International Sleep Surgery Society Research Forum, discussing three main topics, including current evidence for hypoglossal nerve stimulation for therapy for OSA, anatomic and clinical considerations for hypoglossal nerve stimulation therapy optimization, as well as individual factors in the management of hypoglossal nerve stimulation. The stimulation's goal is to open the soft palate and protrude the tongue. Through electromyography needles that are inserted into muscles that measure nerve branch stimulation. To do this, it is essential to understand the human tongue's anatomy. This table here includes the criteria to obtain this implant, focusing on parameters such as age, apnea-hypopnea index, body mass index, central apnea, DICE findings, and whether or not the patient had reacted or had no reaction to CPAP, or continuous positive airway pressure machines. As a result of this panel, it was found that body mass index and complete concentric collapse, or CCC, were found to not be the best for assessing weight impact on success. Female sex was found to be a predictor of the results of the therapy response, and GG, or genioglossus stimulation, was found to significantly decrease mean PURIT. In the future, I plan to have a mentor to help further my understanding of this topic and to narrow it down to a specific aspect of OSA through continuing research. That is my presentation, and thank you.
Our next presenter is sophomore Iman Khalid, who will be presenting about migraines. Hello. Hello, my name is Iman Khalid, and I will be presenting my research on migraines. My main focus this year has been migraines, which was narrowed down from the general topic of neuroscience. Lately, I have been reading articles about different treatments for migraines, both medical and aerobic exercises. To give a little bit of insight on migraines are a nationwide illness that occur in the brain and often attacks one side of the body. It is seen to affect about 18% of women and 6% of men in the US alone. A potential mentor that I've been reading articles about is Daniel DeSouza. DeSouza is an adult neurologist at Stanford University who has many publications on headaches. The ones I've been reading specifically have helped me enhance my knowledge on migraines and different approaches to treat them. The main article I will be focusing on is titled, A Review of Current Evidence in the Surgical Treatment of Migraine Headaches, which, as stated in the title, focuses on the surgical treatment of migraines. The authors include Jeffrey E. Janis, who is also another potential mentor of mine. The main purpose of this article was to find the success rates of the surgery, how long its relief lasts, and what they can do to improve this surgical procedure in the future. This was sparked by the lack of treatments for migraines in the neurological field, even though migraines are highly prevalent and affect many people in both the United States and worldwide. Furthermore, this study was conducted to examine if surgical decompression was a viable approach to treating migraines and should be recommended more in the future. The study consists of 17 different clinical studies, which all consisted of chronic migraine patients who underwent surgical nerve decompression in an attempt to decrease their migraine pain. It is believed that extracranial sensory branches in the spinal nerves can be irritated or compressed at any points throughout the body, which can result in migraines. The reason why researchers are determined to use cell decompression to treat migraines is because it targets these irritated and compressed extracranial sensory branches to ultimately decrease or stop the pain. With this tactic, 4 main sites of the brain are often targeted. The names of these 4 sites in the study are the frontal trigger, temporal trigger, occipital trigger, and the nasoseptal trigger. The researchers created a data table organizing each study, the number of patients and the percentages that corresponded with their total remission, general improvement of their migraine pain, and the brain regions that were treated. The data set found that 68.3% to 100% of the patients throughout all the studies experienced symptoms of improvement. 8.3 to 63.2 of these patients faced total remission of their migraine pain, although these percentages varied heavily due to the difference in each sample size, considering how tiny some of them were. For my future plans, I want to look more into the effectiveness of surgical decompression on a larger population. I also want to focus more on how I can prevent migraines from happening before they actually occur, and I also want to reach out to my mentor and finalize for a mentorship. Thank you.
Our fourth speaker of the night is sophomore Caitlin Lupia, who will speak on the topic of mental health stigmas.
Good evening. My name is Caitlin Lupia, and today I will be presenting on mental health stigmas in men from diverse backgrounds. Mental health stigmas are one of the main reasons why people do not seek professional mental health help. Stigmas are a barrier towards mental health treatment. With that, there are numerous different types of stigma, including self-stigma, associated stigma, and public stigma. Self-stigma is a stigma from an individual towards himself due to due to having a mental illness. Public stigma is from when the public stigmatizes a person from having a mental illness, and associated stigma is where they get stigmatized from a close family or friend. Along with that, stigmas are more common in men due to the standards that they are set to in their masculine norms. With that, self-stigma is the most common stigma that is found in men. Currently, I am looking for a mentor. Dr. David Vogel, a professor at Iowa State University of the Department of Psychology, is one of my potential mentors I sent an email to a couple weeks ago. The article I will be focusing on tonight discusses the masculine norms and the effects on stigma and help seeking in diverse backgrounds. And this article is by my potential mentor, Dr. David Vogel. It was hypothesized that conformity to dominant masculine gender roles will be positively linked with self-stigma and negatively linked with attitudes towards counseling. Positively linked self-stigma it sounds good, but it really isn't good. That means there is more self-stigma in the men. And negatively linked attitudes towards counseling, that means they do not want to seek counseling help. The participants for the study were found on internet websites and also around flyers that were put around villages. The participants were asked to answer numerous of questions on the topics of self-stigma, masculine norms, and their views on help seeking. The age of the study ranged from 18 to 79. Masculine norms have a tremendous effect on men, and especially in men with different diverse backgrounds. In different backgrounds, some cultures accept mental illnesses more and help seeking than others. Attitudes were different based on race and yeah. So this table here shows how everything leads to self-stigma and attitudes. So as you can see on the left-hand side, masculinity and depression lead to self-stigma and the attitudes. In the future, I would like to keep on researching potential mentors and further my research on mental health stigmas and possibly on men. Thank you.
The next presenter is sophomore James Maroulis. He is presenting his research on malware and machine learning.
Hello, my name is James Maroulis, and today I will be discussing my research on malware detection and machine learning. So, malware, as many of you know, refers to the files that try to perform malicious actions on devices like computers. Machine learning is the process of training an artificial intelligence model to perform a specific task, in this case, the detection and categorization of malware. Within the past couple of decades, machine learning has been harnessed to detect malware. However, with how recent of a field it is, there are still many aspects that have yet to be explored. My mentor is Dr. Phil Roth, who currently works at CrowdStrike, which is a computer security company, and he helped develop the Ember dataset, which is a widely available benchmark dataset for training malware detection machine learning models. A key article to my research was Ember, an open dataset for training static PE machine learning malware models. This was written by my mentor Phil Roth and Dr. Hiram Anderson, and it discusses the Ember, which was a novel dataset for training machine learning models to detect malware. The authors, to create this dataset, collected malicious and non-malicious file samples and extracted traits that represented each type of file and labeled these extracted traits to create the dataset. As seen in the graph, there was a dataset for training the machine learning model and testing the machine learning model. Unlabeled samples were included in the training dataset to help see the progress of the model as it was being trained. In the future, I want to continue my research into malware detection using machine learning. I also want to look more into machine learning-based malware classification, or taking malware samples and putting them into a specific malware family. And I would also like to look into the effectiveness of static analysis, or the detection of malicious files before they are run, due to how it was a widely used technique a couple decades ago, but has slowly become more irrelevant. Thank you.
Up next is Parker Molina, who is a sophomore speaking on the topic of agronomics and biochemistry.
Good evening, everyone. My name is Parker Molina, and I will be discussing soil biochemistry and agronomics and the history of my research in this field. Now, before I get started, I would like a quick show of hands from the audience. Now, who here has fertilizer, is preparing to fertilize their lawn for the upcoming spring and summer season? Okay, decent amount of people. We're going to discuss a little bit of how that works. Now, the field of agronomics and biochemistry primarily focuses on the study of agriculture and the soil. And specifically in this case, we'll be talking about how to promote agriculture. Agriculture being the growth of crops. This research focuses primarily on improving soil health while minimizing the negative effects of the usage of fertilizers. Classifying soil health generally includes the measurement of the amount of microbiota and nutrients within the soil, which all have an influence on the generalized health of the soil and the plants within the soil. Now, my mentor is Dr. K.M. Scow, who is a professor at the University of California at the Davis campus. She is part of the Department of Land, Air, and Water Resources, and she has a PhD in soil science and microbiology, which she received from the University at Cornell. She has been employed at the University of Davis for about 30 years now, and I'd like to add she is a very lovely woman. Now then, The hypothesis / the goals of this study is to examine the potential for clover bacterium to colonize the roots of rice plants and to see whether or not this has the potential to promote the bacteria strains of clover, whether or not they have the potential to promote the growth of the rice cultivations. Now, this study was conducted first and foremost by figuring out what bacteria they were working with. So the plasmid profiles of the bacteria were compared. Plasmid profiles being a section of the DNA within bacteria that can be used to easily identify the bacteria that is being worked with. This bacteria was then isolated from the clover plant roots and were grown on agar slopes in order to duplicate the amount of bacteria that the study was working with. And then the population density of the microbiota was measured through the dry rhizosphere mass, the rhizosphere being the area of the soil around plant roots. Now, two different rice seedlings were used, in this case being a hybrid form of rice and a natural variety of rice known as Giza rice. These rice seedlings were grown for 73 days throughout the course of the study, and they were inoculated at about 30 days with the microbiota from the clover. Now, the results. The clover bacteria was found in abundance within the rice rhizosphere. Now, this goes to show that the clover bacteria was capable of colonizing and thriving within the rhizosphere roots of the rice plants. Now, in particular, I would like to point out the E11 and E12 strains of the bacterium, which are notable in their ability to not only stimulate growth, but also within their sheer abundance within the actual rhizosphere of the rice. Now, both of these strains, as mentioned previously, were capable of increasing the amount of grain yield within the rice. Now, if you look at this graph here, you can see the two different varieties of rice used, the Giza 171 and the hybrid IR28. And if you look particularly at the E11 and the E12, you see a significant uptick compared to the control. Now, in this graph, if you look at the highlighted numbers, you will see that compared to the control, the numbers, which is comparing the straw yield and the grain yield, significantly increases under the E11 and the E12 inoculated rice. Now, in terms of my future plans will primarily focus on collaboration with my mentor and ultimately seeing where my research with her takes me. I hope to focus this research primarily on microbiology and its influence on the soil. And ultimately, people, this is the new frontier of agriculture. This is the future of crops, and this is the future of our food and our health. Thank you for listening.
Sophomore Brendan Molnar will now present on his topic of forensic analysis techniques.
Hello, and thank you for coming tonight. My name is Brendan Molnar, and I am currently a sophomore in the Hampton-based science research program. Today, I will be presenting my findings on forensic analysis techniques. As of now, my current research focuses on easier and more efficient crime scene investigation, or CSI investigation techniques, and where there is bias lays in both old and new techniques, and where the room for error in these new techniques are. Additionally, I have been completing bibliographic research on the strengths and weaknesses of traditional CSI and where CSI will be going in the future with technology increase. A potential mentor I have contacted is professional engineer Eugene Lisko, who is a graduate at the Aerospace Engineering program at Ryer Space Polytech University, and founder of AI2-3D, which is a CSI digital reconstruction company where he finds and trains others toward a fully digital forensic finding program. Lisko is a 3D forensic analyst with his main area of expertise being in the future of forensic science as technology becomes more complex. One of my main areas of focus recently has been the 3D documentation and analysis of bite marks, with three-dimensional teeth built onto a computer software, which were directly transferred from traditional dental tooth casts. The study found a stepwise process of teeth impacted, penetrating progressively with increased jaw power, which was previously unknown and thought that constant pressure was used throughout a bite. The initial impression is this necessity to show the accurate comparison in the bite itself, showing the need for speed and efficiency in forensic offices. This study proves the need for bite mark investigation reforms as we progress into the future of technology. It was found In the future, I hope to gain mentorship with Eugene Lisko and continue my bibliographic research into forensic analysis, and to dive deeper into how we can improve our CSI programs with the increase of technology in the future. Thank you.
Our last software presentation will be from Trey Scudder. He will be presenting on behavioral traits of great white sharks.
Hello, my name is Trey Scudder, and I am researching the behavioral traits of great white sharks. So great white sharks are apex predators who are distributed globally throughout the planet. They're a vital part of ocean ecosystems, but currently are vulnerable to being endangered. This is due to a variety of factors, both biological and because of humans. Understanding the behavioral traits of great white sharks is imperative to their protection and management. Currently, there are 2 scientists who could be viable research mentors for me. The first of which being Dr. Craig O'Connell, who was the co-founder of the OC's Conservation Foundation. The second potential mentor is Dr. Taylor Chappell, who is from Oregon State University. An article that has been very useful in my area of research has been the use of baited remote underwater video systems to non-invasively characterize a white shark nursery area off of eastern Long Island, New York waters, and its implications for a potential nursery area. The authors were Dr. Craig O'Connell and others. The study aimed to determine the effectiveness of baited remote underwater video systems, or BRUVs, in identifying a great white shark nursery. BRUVs are used to record data on marine life. They are constructed from PVC piping, and in the study, they were equipped with a GoPro camera, 2 waterproof lights, which permitted nighttime observations, as well as 2 bait cages, which were opposite of the GoPro camera. As you can see, this picture shows 3 of the BRUVs which were used in the study. The BRUVs were deployed opportunistically in 2 regions off of the coast of Montauk, New York, north of Montauk being the Block Island Sound, and south of Montauk being the Atlantic Ocean. Montauk, if you didn't know, is an area that is a white shark nursery. It's been previously identified, and this study attempted to determine the effectiveness of BRUVs in identifying an area as a white shark nursery based on a set criteria. The BRUV's film data was analyzed to see if the area fulfilled all 3 criteria points. These criteria points being, 1, that young white sharks are encountered more frequently in that area than in surrounding areas. The second criteria was that young white sharks remain in that area or return to the area over an extended period of time. And the third criteria was that there's repeated use of the area over several years by young white sharks. The BRUV's data, which was obtained, successfully fulfilled all 3 identification criteria, meaning that the BRUVs could successfully identify the area as a white shark nursery. This is very promising, as it shows that in future studies, BRUVs are a viable method for identifying great white shark nurseries. In the future, I plan on continuing my research on the behaviors of great white sharks. I also plan on connecting with a potential mentor, either Dr. O'Connell or Dr. Chappell, and solidifying a partnership with them. Thank you.
Junior Isabel Ospitali will present the work she has been doing with the guidance of her mentors on piping plover habitats.
Hello, my name is Isabel Ospitali, and today I'll be speaking about the main anthropogenic disturbances and their effect on the piping plover population.
I've primarily based my research focus around the effects of driving on beaches, pet activity, and other different stress factors that arise. I was able to get a more in-depth explanation of how these different aspects of disturbance really affected the plovers on a bigger level than I thought with the help of my two mentors, Dr. Christy Wales in the Department of the Virginia Tech Shore Program and Dr. Chelsea Weithman in the Department of Fish and Wildlife Association. Both Christy and Chelsea are affiliated with the Virginia Polytechnic Institute State University, and I've been communicating with the two over biweekly Zoom meetings and emails. As you can see in the graph, there were less feeding and brooding occurring in the disturbed than the undisturbed, and more sitting and vigilance, which is not very common for the plovers in the day-to-day life and is not good for their population, which causes fluctuation. Many human beach-related activities, such as allowing dogs off leashes, have been negatively associated with the shorebirds' behavior and demography. Where present, dogs are the largest anthropogenic disturbance to shorebirds on beaches. Disturbances such as dogs have been associated with reductions in foraging rates of the shorebirds, which could impair an individual's ability to acquire the energy reserves necessary for future survival or reproduction. In Panel B, the filled circles represent the mean true annual survival for the individuals observed within the study area that breed in the Atlantic coast, which is the black regions, the Great Plains, which is the white regions, and the Great Lakes, which is the gray regions. The dashed lines and error bars represent 95% credible intervals, and the extensions of the 50 to 85%, which is the dark gray, the 85 to 95%, which is the light gray, and the 85 to 100%, which is the black, credible intervals. F represents the proportion of the density distribution that is greater than 0. Furthermore, plovers that experience less disturbance have a higher true survival rate in the Atlantic coast, Great Plains, and Great Lake regions. Chicks are often displaced as a result of disturbance events, which leads to lower access to food and lower fitness. Because of these disturbances, the plover chicks spend less time in moist substrate habitats and spend more time in upland dunes. This led to a large decrease in efficient foraging rates, which widely affected their body conditions. As you can see in the figure, the plovers that experience higher levels of disturbance have a lower mean body mass, which proves the point that foraging habitats, while the plovers that experience less disturbance have a higher mean body mass, which proves improved foraging and brooding rates. Beach closure to dogs and humans have been the traditional but extreme strategy to directly manage this issue, but such a policy is not always feasible due to space, human recreation, public demands and needs, or other pressures. This research has led me to start my research question: how do anthropogenic disturbances such as off-road vehicles affect the piping plovers and their ability to nest and reproduce? This summer, I plan to take trips to Fire Island with Christy and her team in order to take core samples. Core samples are similar to taking a giant cookie cutter of sand to see what invertebrates are in it to determine the distribution of their prey throughout their environment. I will help to collect samples and freeze them so any critters that are in it can't move around, then sift through the sand to get a better look at any sort of plant or invertebrates that are in the sand to be put into a microscope to dissect. This allows the researchers to collect standard sizes and measure the typical amount of prey across the sampled area. It will also help me determine what areas have been exposed to the impact of the off-road vehicles due to sizing and shaping of the sand, shells, and invertebrates that might be avoided by the plovers. I will also be learning how to use dichotomous keys. Dichotomous keys are used when researchers have critters to identify. They first need to determine which key to use, as you can see in the diagram, and research will use those keys to determine different organisms found within the core samples. These techniques that I will learn will make it easier to determine an answer to my research question, as mentioned before. Thank you.
Our next speaker will be Junior Christopher Rowland, who will present his research on marine bioacoustics.
Hello, my name is Christopher Roldan. Thank you all for attending the Science Symposium, and this is my research update for the acoustic characteristics of whales and their implications towards conservation. My research has been a majority of understanding the influences in internal parts of acoustic characteristics of whales as a marine species. A subject to be taken from this is understanding acoustic characteristics will allow for more significant advances in this field, including developing tools for faster and more accurate data collection. The field that I have derived by focusing on is how different kinds of disturbances, such as the ever-rising anthropogenic noise, influences the behavior and physiology of whales. My mentor that is in my current research field is Dr. Manuel Castellet. He has a PhD in psychobiology from the University of Spain back in 2009. He is currently using acoustic tools to study various parts of the physiology and behaviors of cetaceans, including their movement patterns, population distribution, and the effects of intrusive noise. A significant portion of the research that I have been collecting over the past has revolved around the concept of collecting acoustic data that is used for research. There exist several methods for collecting such data. One method includes using devices called hydrophones, devices capable of accurately receiving acoustic signals underwater. These devices can be placed on boats that can patrol in a certain pathway, or more commonly, placed in certain locations in what is known as passive acoustic monitoring, or PAM. PAM is when hydrophones are placed in specific conditions so that they can receive signals over a prolonged period of time, typically recording for years or decades. Another method that can be used is archival tags, which can record specific whales in addition to providing additional information that can be received by satellites, set usually locations on the globe. From extracting the recordings gathered by the hydrophones and archival tags, these can be placed into spectrograms to reveal two different types of calls that can be emitted by whales: upcalls and shots. Upcalls are the sounds that one would typically hear during whale watching if you decide to go in a whale watching tour somewhere on Long Island, like Port Jefferson. Shots, however, are short, impulsive sounds that whales emit during impulsive or social behaviors. These spectrograms were provided from a study done by Dr. Parks and Dr. Matthews in 2021. Although these graphs do not display the same scale of time or frequency, these graphs do display accurately the characteristics of both upcalls and shots. Upcalls are generally lower sounds made for a longer period of time, typically up to 1 to 3 seconds. And shots are generally higher sounds made over a minuscule amount of time, less than a second, as seen on the graph on the right. As discussed earlier, these kinds of sounds and data can be derived from a wide variety of methods, including underwater recorders and attachable tags that can be placed on the whales themselves. It is important to obtain such information as to recognize when a given anthropogenic noise would be too intrusive for the acoustic capabilities of whales. In the same study conducted by Dr. Parks and Dr. Matthews, a table was provided through them that showed the ranges of the recordings extracted for their study. The upcalls were as expected, with the minimum and maximum ranges being significantly lower than those of the shots, which were much shorter compared to the upcalls. Another significant portion of my research includes what the information that we can derive from records and tags contributes to actual conservation efforts to preserve endangered whales. From the acoustic information that we can extract from whales, we are able to accommodate the whale's abilities from this acoustic information via imposing limitations on the intensity of human motor equipment or the knowledge of where exactly a species of whales is at any given point in time. One aspect for how obtaining acoustical information for how it can contribute to conservation efforts is the ability to speculate a whale's distribution pattern through mass acoustical recordings. In a study developed by Dr. Baumgartner and others in 2020, the team used various hydrophones across the Atlantic coast from Florida all the way down to Maine to determine the seasonal distribution of various types of whale species. These recordings were undergoing various durations of passive acoustic monitoring. As seen in the graph, the black dots represent those that were recording between 2011 and 2014. The yellow dots represent those that were recording between 2004 and 2010. And the blue dots represented those that were recorded for both periods of time. One notable factoid that was derived from various experiments is that while all the whales have the exact same templates of calls used in different situations, that being up calls and shots as referenced in the top left, each species of whales has their own unique frequency range that they tap into when emitting these calls. These maps were all taken in the various winter months from the years of 2004 to 2014 in the same study. Each graph representing one kind of species that has traveled to a specific area of density along the coast. The pink dots on the left represent the various locations fin whales travel to, the middle brown dots represent the locations of blue whales, and the rightmost green dots represent sei whales. These locations allow for the knowledge of where a species would be present in times of conservation, making the process less arduous. This progress that I have made with my research has led me gaining the inspiration to pursue a research question of my own. How can we accommodate the acoustic characteristics of whales around Long Island to ensure a prosperous environment for both humans and whales? The future plans that I hold for my research include driving my research in the direction of pursuing information regarding the whale populations around Long Island, and the interactions between those intrusive noises to the physiology of whales. I will also use the RAVEN program to progress my research within these areas. RAVEN is a program provided by Dr. Castellet, my mentor, which allows for the analysis of any type of recording that you are able to input into the program, including various forms of waves, spectrograms, and most importantly for my research, exact frequency values for any selected part in the recording. Thank you for your time, and we hope you enjoy the rest of our science symposium.
We will now move on to our senior student speakers. Our first speaker will be Dylan Foley, who will present his work on horseshoe crab movement.
Hello, my name is Dylan Foley, and this is my presentation on horseshoe crab habitats and movement. But before I get into the details of my study, let me quickly mention my mentor, Dr. Scafani, who's with us today. Dr. Scafani is an ecologist that is a part of Cornell Cooperative Extension on Long Island and who studies horseshoe crab conservation and behavior of horseshoe crabs. He runs the Horseshoe Crab Monitoring Network and served as a coordinator for the Peconic Estuary Partnership. He has also helped guide me through my research process with bibliographic and field research. He has also introduced me to Dr. Justin Bopp at Massachusetts Division of Marine Fisheries and Stephen Havens from Cornell Cooperative Extension. Without them, this project would not have been possible. My research question is, how is juvenile horseshoe crab behavior impacted near beach spawning habitats based on prosoma size and sex differences? The hypotheses created are distance will increase with horseshoe crab size, the overall space used by juvenile horseshoe crabs will be different between the spring and fall months, and males and females will travel similar total distances. This is important as larger animals often move farther distances, and trawl surveys in Delaware Bay indicated that larger juvenile horseshoe crabs had a greater presence on the continental shelf compared to estuaries. Juvenile stages of horseshoe crabs range in size from 1 centimeter to around 170 millimeters before reaching maturity. While a lot is known about the behaviors and habitat use of smaller juveniles and adult stages, very little is known about the intermediate size ranges of juvenile horseshoe crabs between 90 and 170 millimeters in size. This stage is important because they spend a considerable amount of time at that stage and may be vulnerable to human activities. It is therefore important to understand their behaviors and habitat use during this stage. So, why are horseshoe crabs important? Horseshoe crabs are extremely important in the biomedical industry. Their blood is important as the limitless amoebocyte lysate test is a great indicator for the presence of gram-negative bacteria. Their blood is able to detect as little as 1/1,000,000th of a billionth of a gram of endotoxin, making it extremely useful for the detection of pathogens in vaccines. hence why pharmaceutical bleeding has increased since COVID-19. There are 5 biomedical industries on the Atlantic coast of the US that bleed horseshoe crabs. Horseshoe crab bleeding affects their movement and survival. On top of this, commercial fishing is a method used to provide bleeding facilities with the crabs. Commercial fishing also uses horseshoe crabs as bait for American eel and conch fisheries. Crab eggs are also function as a food for shorebirds during their migratory season. The shorebirds double their weight in 2 weeks and consume as many as 135,000 eggs. Loggerhead turtles are also main predators of the horseshoe crabs, but many hermit crabs were found preying on the developing horseshoe crabs. As a result of these stressors, management of horseshoe crab spawning location is needed. Although some habitat conservation programs can pose a threat for horseshoe crabs through erosion control, such as dredging, creation of bulkheads, as it may destroy spawning and nursing habitats. Management ideas such as the programs that involved controlled harvests are put in effect for population protection. Spawning surveys are also conducted to help determine and conserve horseshoe crab population, as little is known about the entire population. Data collected from these surveys could be used for management of habitats. To better collect data on horseshoe crabs, acoustic tagging has been used to study how these stressors impact the species. It is believed that acoustic and radio transmitters can make studying horseshoe crabs in the foreshore and inshore easier. Telemetry, a process of obtaining data remotely by transmitting information from a marine animal, could help with tracking over a spatial and temporal scale, making it effective and accurate. The tags send an acoustic sound signal that is detected by an acoustic receiver placed on the seafloor. Data collected from horseshoe crab spawning can be used along with surveys to improve beach management. Acoustic telemetry is not just used in horseshoe crab research, but in research involving predation by whelks on bay scallops or whale behavior. Similar to horseshoe crabs, telemetry can be used for turtles, as it is necessary for the conservation of a threatened species. A popular field of use is in shark studies. Shark studies are one of the first to use telemetry on marine animals, as it is necessary to understand their interactions with humans. Telemetry is helpful as it can indicate what animals are near telemetry receivers and what they are doing. The juvenile study involved collecting horseshoe crabs on the sandbars of Murchis Bay. The image above shows the map of Murchis Bay with radio receivers shown as gray crosses. 32 acoustic receivers were used to monitor fine-scale movements of tagged horseshoe crabs throughout the bay. This method triangulates the position of tagged horseshoe crabs every 2 minutes. There were 19 crabs detected in the fall and 38 in the spring. The different colors showed the movement trails of the tagged horseshoe crabs. After we collected the crabs, we went back to fill out the data sheet with measurements and the tag ID of the horseshoe crabs. We attached a conventional tag pin on the horseshoe crab and then went through the acoustic tagging process. The conventional tag is similar to the tags attached to birds, with a barcode and a bunch of numbers. Acoustic tagging is used to pinpoint a marine animal's location. This process involves sanding and cleaning the prosoma of horseshoe crabs, followed by glue and accelerant. The glue and accelerant created a chemical reaction to hasten the gluing process. These crabs were then released. 25 horseshoe crabs were released in the fall and 25 in the spring. The prosoma was cleaned with ethanol, dried with a painter's rag, and then sanded down. We used glue with glue accelerant to attach the Velcro and tag on the prosoma. The reaction is exothermic, releases heat, so we were told to apply glue generously, but not too much where we fry the brain, as the neural network of horseshoe crabs is below the surface of the prosoma. The object on top of the horseshoe crabs with the red tape is the acoustic tag. The white disc is the conventional tag, and in the far right image, I am cleaning the prosoma of an adult horseshoe crab. All images above are the tagging of an adult horseshoe crab and not a juvenile. The data was run through our software coding to work on downloaded receiver data to create readable data and graphs. The Memento HMM-R package was used to assess movement paths of the crabs. The package also includes covariates, such as size and sex of the individuals to model factors that influence their movement. The plot shows movement of the different sexes between spring and fall. Red represents the females and blue represents the males. Both plots show a greater movement by males than females. The graph here shows the qualitative analysis of spring juvenile horseshoe crabs positions with a fine-scale receiver array in the spring. The receiver locations are shown with black dots. The ID numbers correspond with the color, and the splatter of color shows their movement. The plot shows that all individuals in the acoustic array avoid the top left area and are very spread out. The plot here shows a qualitative analysis of juvenile horseshoe crab positions within the fine-scale receiver array for the fall. The plot shows a contraction of habitat use and still somewhat avoiding the top left corner. These scatter plots both represent distance covered based on prosoma width. The left plot for spring shows that distance is decreasing with an increased prosoma size for horseshoe crabs. However, the plot on the right, representing fall, shows more scattered dots. The p-value for the data test, which determines how statistically significant results will be, shows that the data was not non-significant at 0.38. So, how is juvenile horseshoe crab behavior impacted near beach spawning habitats based on prosoma size and sex differences? This brings me back to my hypotheses that distance will increase with horseshoe crab size. The overall space used by juvenile horseshoe crabs will be different between spring and fall months, and males and females will travel similar total distances. Although we reject the size hypothesis for spring, we fail to reject it for fall, meaning travel distances were related to horseshoe crab size in spring, but not in the fall. We also found differences observed between spring and fall habitat use. Some areas were avoided more so in the spring than the fall. Despite t-test results not statistically significant for gender data, there was still greater movement in males than females. This is the first study to investigate the behaviors of juvenile horseshoe crabs. In addition, acoustic telemetry was very effective at tracking their fine-scale movement within the bay. It is important for the conservation management and habitat protection from activities such as dredging. There needs to be continued research on developing behavior movement models to quantify and predict their behaviors, as this was preliminary data. So, my future direction in college, I'm pursuing a major in business management. I finally made a college decision this week very recently. I made a decision to continue my academic journey at the University of Georgia. Although I'm leaving the field of marine biology, I do hope to continue my research into different topics and utilize my abilities in college. Thank you for listening to my presentation, and thanks again for my mentor.
Our next presentation of the evening will be given by Senior Emma Halsey, who will be discussing her research on marine cold stunning.
Hello everyone, my name is Emma Halsey. Today I'll be presenting my research topic on marine cold stunning, specifically on 3 sea turtle species, the green, loggerhead, and Kempsford turtle. I've been researching this topic for the past 3 years of science research, as my freshman year topic started off with research on coral bleaching. I decided to choose the topic of cold stunning due to this issue being a common problem focused on in New York's coastal waters. Before I get into my specific research, I wanted to take a minute to thank my mentor, Dr. Griffin, for his tremendous support and help towards my research. In July of 2022, I established a mentorship with Dr. Griffin, who is affiliated with the University of Massachusetts Amherst. He specializes in environmental biology, ecology, and conservation research, in which his personal accounts and experiences of conservation work with marine life has given my research a better understanding with outside knowledge. He has also provided me with numerous peer-reviewed articles, some of which are his own, that have supported the development of my research studies. The main focus of my research explains the huge issues that has contributed to the increasing mortality rates in marine habitats. Due to temperature change in the water and atmosphere, beach strandings occur that cause species to become lethargic and vulnerable to their surroundings. A hypothermic-like state develops for these animals from decreasing water temperatures, particularly once reached below 10 degrees Celsius and 50 degrees Fahrenheit, that affects their inability to migrate out of seasonal waters in time for the winter months. In recent years, the number of marine impacts has severely heightened, causing health problems and the inability to swim out of these oceans. This graph shown here represents the number of individuals in occurrence time of cold-stunned sea turtles in each winter with respect to water temperatures. The bars represent the number of turtles encountered, and the lines represent surface water temperatures at a monitoring station. Rising sea surface temperatures in specific areas around the world cause migration and movement patterns to shift. I have learned that hypothermic-like behaviors could be caused by rising sea surface temperatures, climate change, and just not having enough time for migration. Water temperatures are dropping way too quickly as sea surface heightens, which causes sea turtles to get stuck in colder waters. Throughout my research, I have focused on 3 of the 7 sea turtle species, in which one in particular has shown up most prominently from strandings. The increase in cold stunning events of Kemp's release is largely influenced by the determining factor of their ecology. Smaller juveniles will feed in nearshore areas to grow and develop. However, warming sea temperatures cause movements further north, which means they're utilizing the North Atlantic Ocean more than they previously did. Changing migration patterns also enlarges the likelihood of being caught in natural catchment areas that is detrimental to Kemp's release, who make up a small size class based on their shell length and, again, their ecology. Specific locations these turtles make up include New York, New England, North Carolina, the Gulf of Maine, and the Gulf of Mexico. My research has mainly focused on areas such as Long Island Sound and Cape Cod Bay, but has extended through article research. Species such as the loggerhead, along with the green turtles, have also been a crucial species found in local New York waters, in which these strandings have commonly shown to affect turtles on shorelines and north-facing beaches. Exploring the cases in the Northwest Atlantic was significant to my research due to these areas being so closely located to me. With the help of Dr. Griffin, we developed the research question, how does cold stunning affect differentiating sea turtle species in coastal waters, specifically the Kemp's Ridley, green, and loggerhead species? There are predictions as climate change has an influence on the increasing cases relating to sea surface temperatures, as this has shown again to continuously increase. The target response to cold stunning is health impacts of a lethargic body state, dehydration, infection, and possible mortality. These possible responses promote strandings where immobility causes the individuals to wash up on nearby beaches. Ocean currents, tides, and wind-driven patterns influence a buoyant state that drives the turtle into the shore to strand. This problem as a whole has been around for hundreds of years, but has only more recently become a concerning issue. Along with New York, Cape Cod Bay is another prominent location, as mentioned before, where cold stunning is common. This issue can also be influenced by geographic factors, including natural catchment areas. The bay has had a large number of cases due to the bay being semi-enclosed, with an increase in Kemp's Ridley cases in the bay due to warming sea surface temperatures in the Gulf of Maine. Here's a graph of the backtrack positions of stranded sea turtles in 2012 and a 2013 case. The red dots show the landing locations in November and December, and the blue and green dots show the estimated locations of backtrack turtles that stranded in, again, November and December, when they first encountered water less than 10.5 degrees Celsius. These estimates assume that passive or non-swimming turtles were only transported by currents when modeled surface temperatures were less than 10.5 degrees Celsius. The yellow and magenta lines are examples of backtracking turtles that are stranded in the westernmost and easternmost locations that suggest that these turtles were first cold stunned near mid-Cape Cod Bay. Fortunately, conservation efforts and volunteer influences have allowed for an alternative impact for health concerns. The rehabilitation process consists of treatments of warming for dehydration and conditions such as pneumonia that commonly come up in stranded patients. Through exams, conservation and/or rehabilitation centers will determine the current state of the animal and whether they can survive in the wild or not, through treatment plans that are given to each patient. Most rehabilitation methods consist of using blood samples to conclude their specific health status. If treatment plans are completed successfully, that affected sea turtle has possibility of release, but if the patient does not meet the release requirements, they are most likely kept in an aquarium or sent to stay at a conservation center. Survival of species through rehabilitation efforts have been critical due to tremendous attempts to reverse this overall problem. As mentioned before, the Kemp's-Ridley species has been most commonly focused on in research efforts, as they are in the most crucial conditions. The Northwest Atlantic area's weather is unable to be predicted, making it harder for migrations in inhabiting this area. Cape Cod Bay has been an influential location for cold stunning that affects swimming abilities and movement patterns. There has been a hypothesis that after a cold weather event, sea turtles become either buoyant from gas buildup or become immobile due to dive movement towards the bottom. Specifically for green species, imbalances in blood chemistry and metabolic pathways have been affected by hypothermic events, causing flotation towards the surface. Leatherbacks are another one of the common species found in New York's waters, but have been excluded from this research because they are not as drastically endangered compared to the other three prominent ones focused on here. Throughout this research, there are still unanswered questions surrounding this topic, such as what happens to the patient after release. Questions range from whether or not rehabilitation is 100% effective, or if released turtles could repeat cold stunning again. There's still little to know about the aftermath of release and predictions of whether or not warmer water temperatures actually created the strandings events from the drastic shock change. Here's a graph I developed referencing the data collected from the peer-reviewed article that studied the historical nesting levels of Kemp's-Ridley sea turtles and then compared them with current years. This study was influenced by the Herrera film of 1947 based on the mass nesting event. As you can see, there was an increase in number of nests, but is now decreasing as the graph gets closer to 2014. There's also an irregular pattern of percentage data that suggests numbers could change randomly when unexpected. Researchers are still questioning whether populations will recover to historical levels with the current percentages of nesting trends. The survival status of an endangered sea turtle population is the estimation of the historical numbers of nests prior to their decline. Continuing with the information from the graph, from 1947 to 1985, the population of Kemp's-Ridleys went through a decline of 99.4%. Since the decline, there has been a slight recovery, but has more recently started to change again, with current levels of nesting being 9.9%. During the 1947 mass nesting event, there were 26,916 nests recorded and a predicted 121,517 total nests for the season. This number of nests declined from 121,517 in 1947 to 702 in 1985. This data covers the historical nesting population of species that can be compared with cold stun count, as these numbers are predicted to again continue to increase. This is another example of future predictions of case numbers. The study believes that as sea surface temperature increases in the Gulf of Maine, by 2031, at least 2,300 Kemp's-Ridleys will be cold stunned per year. These events at lower latitudes are caused by cold weather, while events at higher latitudes are caused by low water temperatures from species not migrating south early enough. Warming sea surface temperatures in the Gulf of Maine may have caused Kemp's-Ridley's neritic developmental grounds to change towards the north by the Atlantic coast. These numbers will continue to increase if the Gulf of Maine's warm temperatures push the animals into nearshore waters. Sea surface temperature and hatchling number was the common cause of events with Kemp's-Ridleys, with cold stunning events being more common when sea surface temperatures was warmer during the fall months. There are suggestions that the Kemp's-Ridleys make up New York's waters in early developmental stages. Along with Long Island, New York, Kemp's-Ridleys commonly migrate and distribute to the Gulf of Mexico and areas near New England. During changing seasons in northern areas, species such as the green and loggerhead will protect themselves from declining temperatures by hiding themselves in sediment at the bottom of the ocean. Mortality rates were shown greater than 75% in cold-stunned turtles, with the majority of this percentage being made up by the Kemp's-Ridleys. It was shown that water temperatures were uniformly distributed in the water column, as there were t-tests used to show there was not a difference at the surface compared to the bottom of the water. Most of the cold stun turtles, all juveniles, were found in shallower waters with temperatures dropping below 10 degrees Celsius. The Ridley's mean size was found to be smaller in New York's waters than any other area based on their carapace measurements. Lastly, the research concludes that all 3 species are determined to be endangered with many threats from their surrounding areas, but the Kemp's Ridley has, again, extensively concluded to be most impacted compared to the green and loggerhead, who are still largely impacted. Lastly, this past February, I committed to the University of Rhode Island, where I plan on possibly continuing research at the university in the fall. There's a coastal environmental program there with internships with the NOAA on their Bay campus. This will give me an opportunity to continue my research into college that ties in with my environmental science and management major. This concludes my presentation. Thank you so much for listening.
Presenting next is Senior Samantha Kraykar, the salutatorian of the class of 2024. She will be presenting her senior research project on cancer metastasis.
Hello, my name is Samantha Kraykar, and my research topic over the past 4 years has been the metastasis of cancer, which is the dissemination of cancer from the primary tumor to distant parts of the body. Cancerous cells are then able to form a new tumor in healthy organs or tissues throughout the body. During this process, cells lose their polarity and severs intercellular adhesive junctions, thus becoming a migratory mesenchymal cell. This process is known as epithelial to mesenchymal transition. During my sophomore year of this program, I established a mentorship with Dr. Gabor Balazi, who is the head of a research lab at Stony Brook University. There, he performs research regarding gene circuits, which they use in an attempt to communicate with cancerous cells in an attempt to make them behave better. He has sent me countless articles that I use to begin my sorting and worked to contribute into my research paper. We meet every few weeks to discuss progress regarding our research, data collection, and findings. Many of these articles I found most useful to develop and answer my research question, which I will get to in a minute. To establish a baseline in my chosen field, I found around 17 to 20 articles that largely contributed to my research in order to fully and effectively answer my research question. It is known that cells with different epithelial and mesenchymal phenotypes can exit the primary tumor, with epithelial cells exiting either through EMT transition or by other mechanisms that allow cells to be released as single cells or in clusters. The capacity of cells to interconvert between phenotypes is wide and encompasses both epithelial mesenchymal and mesenchymal epithelial. At secondary sites, solitary cells or clusters may acquire dormancy before resuming their growth. With all of that being said, there is an urgent need for new markers to define different stages during this transition between these phenotypes, as well as into their dormant state. There have been a growing number of studies that analyze the EMT process being specifically targeted for therapeutic benefit. This brought me to my research question: how does genetic manipulation of cancerous cells determine the amount of therapeutic agents in relation to epithelial to mesenchymal transition? While EMT has been regarded as a phenotypic conversion between cells, recent evidence indicates that it plays a critical role in stemness, metabolic reprogramming, immune evasion, and therapeutic resistance of cancer cells. EMT induces cells to suppress epithelial features, turning to mesenchymal expression and aiding invasion. This can be seen as a plastic program as well, in that cells reversibly and dynamically alter their molecular and functional traits along the epithelial to mesenchymal spectrum. Since EMT is a new topic and not much is known about the behavior of these phenotypes in regards to genetic manipulation, I found my research would have the most impact being made into a lit review, analyzing previous research through a different lens. There are a few variables to my research question that I needed to specifically address through these articles, such as genetic manipulation in regards to tolerance and resistance to treatment. In monitoring EMT genes, we are able to better predict tumor response. In my research, I also found it beneficial to include metastatic properties in regards to cell behavior, such as cancer stem cells, aka CSCs, which are undifferentiated cancer cells exhibiting stem-like features. The emergence of these CSCs occur as a result of the EMT process and leads to invasive behavior. While I was focusing on different therapeutic techniques for patients with disseminated cancer, I chose to exclude my research regarding driving factors of important driving traits, such as the mitochondria tumor repopulation and cancer stem cell maintenance. Different mechanisms continuously adjust their architecture, position, and overall fitness in response to cellular and extracellular cues. Although this research is pivotal in the field of metastasis, I found it irrelevant to my research. This image depicts an analysis of EMT expression in cancer stem cell-related proteins, where the first, second, and third columns show non-neoplastic mucosa, the tumor center, and the invasive front, respectively. The epithelial markers are the surrounding dark red and dark red-purple colors, while the mesenchymal markers are the lighter colors towards the center. I have spent numerous hours analyzing and interpreting images like this in order to form correlational data sets and graphs. After analyzing a number of treatments, I was able to filter out ones directly linked to genetic manipulation. Gene transfer is a new treatment that introduces new genes into a cancer cell or the surrounding tissue to cause death or slow the growth of cancer. This treatment technique is very flexible, and a wide range of genes and vectors are being used in clinical trials with positive outcomes. It has been established that DNA changes cause cancer. However, since these are more recent findings, this topic needs to be further researched to establish effective methods to address this. Tools such as the CRISPR have become a new mainstream technology, working in that in order to protect themselves against invaders like viruses, microbes capture snippets of the invasive cell's DNA and stores them in a way in segments called CRISPRs. This can be done either through the EMT transition or during either definitive state. If the same cell tries to attack again, those DNA segments help an enzyme called Cas find and slice up the cancerous DNA. As my 4 years of research and high school come to a close, I would like to thank Ms. Lavazzoli, Dr. Forsberg, and most of all my mentor, Dr. Gabor Balazzi, for all of their support along the way. I would also like to thank Ms. Fassini for putting together this symposium so that me and my classmates can present all of our hard work from these past few years. Next year, I plan to continue to pursue my passion for science at CCNY studying biochemistry. Thank you for listening. I hope you all have an amazing night, and I wish the best to the underclassmen and where their research takes them.
Senior Cassidy Liparati will speak next on her years of research into relapsing remitting multiple sclerosis.
Good evening, everyone. Today I'm going to be discussing whether sex reveals any differences or patterns in the occurrence of adverse events or AEs for multiple sclerosis or MS patients undergoing fingolimod treatment. I developed my topic with the help of my mentor and vascular biologist, Dr. Law, who is affiliated with Harvard University and the Boston Children's Hospital. He has his own laboratory through the Boston Children's Hospital, the Law Laboratory, where he studies the biology of sphingosphene 1-phosphate, or S1P, which will be explained later in my presentation. My mentor has met with me over Zoom, helping to explain topics, and has answered any questions I've had, overall making this research doable for a high school student, and I'm extremely appreciative of the time he has offered me. He has helped me to narrow my topic from researching plant metabolites to fingolimod for MS specifically. Multiple sclerosis, or MS, is a disease of the central nervous system where lymphocytes or white blood cells cross the blood-brain barrier, or BBB, directly disrupting and causing inflammation within the central nervous system. This causes damages to our nerve cells, and nerves are important because they allow for our body to communicate and for us to react properly. The image above shows, The image above shows a nerve cell and what may result from MS, including demyelination, which myelin would normally help to speed up transmissions along nerve fibers, and it also helps to protect the axon, and now without it, further axonal damage may result, and this is damage to the part of the axon that actually carries signals to and from the body, and this axonal damage causes permanent neurological impairment. Relapsing and remitting multiple sclerosis, or RRMS, is the focus of my research, as well as the most common form of MS, making up about 85% of cases. This form consists of periods of relapses or an outburst of symptoms followed by periods of remission. I'm focusing on this form because the treatment I am focusing on was approved for RRMS. With this form, for every 3 female patients found, only 1 male patient has the disease. Clearly, women are disproportionately affected by the disease, explaining why females are predominantly included in clinical trials evaluating potential therapeutic effectiveness. As a result, there lacked data on men compared to women, and that was one limitation I had run into. The therapy I will be focusing on for my research is Gilenya, or its chemical name fingolimod. Fingolimod was the first oral drug approved for relapsing forms of MS by the FDA in 2010, a treatment option preferred by many. Fingolimod is an immunosuppressant developed from a natural product, myriocin, or ISP1, a metabolite of the fungi Azaria sinclairi. The compounds myriocin, sphingosphene, and fingolimod are shown above, and major similarities can be seen among these compounds. Fingolimod is a sphingosphene 1-phosphate, or S1P modulator, meaning it targets S1P receptors found on various cell subtypes throughout the body, and these receptors have adverse functions that give the drug medicinal properties. Fingolimod's main function is achieved through targeting lymphatic or S1P receptors on lymphatic organs, which allows, it prevents the normal function of S1PRs, which would have allowed for lymphocyte egress, or the release of lymphocytes, and this in return reduces lymphocyte presence in the blood. Fingolimod is very effective due to its lipophilic ability to cross the blood-brain barrier, or BBB, and actively work on the central nervous system directly. The blood-brain barrier prevents toxic substances and compounds from entering the brain and allows good substances to enter, such as glucose or O2, or in this case, fingolimod passes due to its size and partial lipophilic structure. Fingolimod has been shown to reduce relapse rates by approximately 50%. Efficacy in reducing relapse rates has been shown to be generally equal in the studies performed between females and males. However, differences in terms of sex may appear when evaluating adverse events, or AEs. Little is known about sex differences in drug metabolism within the gastrointestinal tract shown above, and this is the passageway of the digestive system, including the organs involved in digestion and the processing of foods, or in this case, a drug. As an orally administered medication, fingolimod likely faces differences at this level, which is going to have major impact on drug delivery and bioavailability. Further differences include women having a longer drug elimination time, which has a relation, though not fully understood, to unwanted side effects. This is likely due to differences in liver or hepatic enzyme activity. Biological differences between sex is contributing to differing drug elimination abilities and delivery abilities, showing a potential for adverse events to occur and the lack of understanding surrounding this. Although the therapeutic goal of fingolimod is to redistribute lymphocytes as an immunosuppressive drug, this is also associated with lymphopenia. Lymphopenia occurs when your body doesn't have enough lymphocytes or white blood cells to fight or protect against infections. Low lymphocyte counts have been shown to increase the risk of infection in those with lymphopenia. Although less severe forms of lymphopenia are expected, a lymphocyte count below 200 cells per microliter of blood, which is complement with grade 4 lymphopenia, resulted in treatment discontinuation. There are 4 grades of lymphopenia, with grade 4 representing the most extreme of cases. I have chosen to focus on lymphopenia as it is the most common adverse event associated with fingolimod. Studies have shown altering results in the risk of occurrence, with one study reporting only 5 out of 358 patients, which is only about 1% with occurrence of lymphopenia, and another study finding this percent to be 15 to 20%, which is obviously very different. Further, data is minimal in terms of sex, which brings me back to my question. Does sex affect the risk of grade 4 lymphopenia among RRMS patients undergoing fingolimod treatment? The goal of this question is to help to shed more light on the safety of treatment options and to help to individualize therapy option. I hypothesize that there will be a statistical difference in the occurrence between females and males. I'm performing a meta-analysis, so I have incorporated the results of different articles that have evaluated medical records from hospitals and referral centers. Patients included were diagnosed with RRMS, were receiving 0.5 milligrams of fingolimod, which is the FDA-approved dosage, and data was provided individually for females and males concerning occurrence of lymphopenia. Further, grade 4 lymphopenia, which is a lymphocyte count below 200 cells per microliter of blood, as mentioned, defined fingolimod-induced lymphopenia. As mentioned, I am performing a meta-analysis, which consists of me adding the results of a variety of articles in order to evaluate trends in a large pool of data and draw stronger conclusions than the individual studies could. With the data selected, risk of occurrence was determined for both men and women. As mentioned, a limitation I faced was it was much easier to gather data regarding women, so I attempted to gather as large of a cohort of males as I could. Fractions revealing risk of occurrence for each study was added together to get a total, and I performed the t-test on the combined data, investigating whether there is a statistical difference in occurrence for men and women. The t-test is a statistical tool used to confirm whether or not there's a statistical difference, in this case between females and males. This result will confirm or reject my hypothesis, showing if data broken down by gender was necessary or made no difference in each population's percent of occurrence. I finally created a graph similar to the one shown above, which I found from an article with similar goals as to my own research. And as you can see, it shows the percent of population experienced the occurrence broken down by males and females. However, my graph will relate to lymphopenia specifically, the adverse event at focus. I've also created another chart revealing baseline characteristics, and this has the goal of making data on the matter more accessible. I have found there to be a statistical difference in the incidence of lymphopenia between men and women, supporting my hypothesis. Women faced a 16.95% risk, while men faced a 10.61% risk. I have arrived at a p-value of 0.0054, which is being compared to 0.05 or 5%, with values below showing a significant difference in occurrence. As you can see, this value is nearly 10 times smaller than the 0.05 value, showing significance. In previous studies, similar results had been seen, but significance wasn't able to be proven in those cases due to the smaller sample size. It wasn't able to be applied to the overall population as easily or accurately as the larger cohort I have now gathered. My result shows the importance of looking at adverse event data in terms of gender, not just for fingolimod, but for therapies in general, as these differences are likely due to differing elimination abilities among sex as well as immune responses. In addition, ethnicity should also be examined, as similar to sex, ethnicity has shown to have an effect on drug eliminating abilities. Other adverse events should also be examined for fingolimod, with infection being an important adverse event after my findings, considering lymphocytes are what help to fight infection, and now without them you would expect infection would increase. But interestingly, one study does find the opposite to be true, and they find there to be a relationship but not a significant correlation between lymphocyte counts and infection during fingolimod. But other studies prove the opposite to be true and show that there is a correlation between lymphocyte counts and infections, showing a need to further investigate this. In addition, other adverse events not related to lymphopenia must also be examined for fingolimod and other therapies, as trends will likely continue due to biological differences between sex. I'm still contemplating my final decisions, but I would like to pursue my passion for biomedical sciences beyond high school, whether that be at Stony Brook University or SUNY New Paltz, both strong universities not too far from home. Either way, I'm happy for the opportunity to pursue my interests beyond high school. Before I conclude my presentation, I would like to take a minute to thank everyone who has supported me throughout my past 4 years in the program, including Dr. Forsberg and Ms. Lavazzoli, who have helped us to ensure we've had all the means to complete this program. And I would also like to thank Ms. Vicini, who has put in a lot of time and effort to make tonight happen, as well as my mentor, Dr. Law, who has offered me a lot of time and support. And I would also just like to thank Dr. Z and Mr. Garvey, who have stepped in and helped me to finalize my research project. That concludes my presentation. Thank you for listening.
Our next presentation will be given by Senior Manal Naeem, who will be discussing her work on cardiomyopathy. Hello, my name is Manal Naeem, and today I'll be discussing multiple imaging modalities in assessment of hypertrophic cardiomyopathy. Here you can see an extracellular volume image, which I will be getting into later on, but as you can see in this image, the volumes are represented by the different colors. Before I introduce my research, I'd like to introduce my mentor, Dr. Warren Manning, who is affiliated with Beth Israel Deaconess Medical Center and Harvard Medical School. He specifically focuses on cardiomyopathies and other cardiovascular diseases and uses imaging modalities such as echocardiography and cardiac magnetic resonance. We established a mentorship in May of 2022, and I've been fortunate enough to meet him in person. He's helped me greatly throughout my research and has helped me develop my topic. He's sent me many peer-reviewed articles and worked on my research paper with me, as well as helped me produce my poster and slide presentation. I am very grateful to have him as a mentor, and I'll never be able to thank him enough for the amount of help he's provided me in my project. Hypertrophic cardiomyopathy is a genetic disease that leads to hypertrophy or thickening of the wall of the heart. Usually this occurs in around 1 in 200 or 1 in 500 people. Although you may think that this isn't significant, over a certain amount of time as a hypertrophy develops, it can lead to worsening outcomes, the worst being sudden cardiac death. This is usually caused when there are increased interactions between myosin and actin, which are protein filaments. Usually, the interactions between these proteins lead to contractility of muscles, but when there are increased interactions, then there's hypercontractility. This hypercontractility leads to formation of hypertrophy in the heart. This is a genetic disease, which is why many people undergo genetic testing if they're aware that someone in their family has this disease. If people are unaware that someone in their family has this disease, or this disease shows up as asymptomatic, it's important to undergo imaging to assess for this disease. There are many imaging modalities that can be used to characterize hypertrophic cardiomyopathy, which includes cardiac magnetic resonance and cardiac ultrasound, which is also known as echocardiography. These are the two mainly used methods as they are readily available and do not cause any harmful ionizing radiation to the patient. There are many subtypes of CMR, which includes T1 and T2. Both of these methods allow for tissue characterization of a patient with hypertrophic cardiomyopathy. T1 specifically looks more at fibrosis, whereas T2 looks more at inflammation. This image here is a T1 of a patient with hypertrophic cardiomyopathy. As you can see here, the patient has hypertrophy, and the different colors show how long it takes for the patient's heart to return back to its normal, to return back to where it started from. Extracellular volume was an image that was shown at the beginning of the presentation, and it's quite similar to this method, although that specifically looks at the change in volume of a patient who has this disease. There's also cine imaging, which is a sequence of images that's taken over a certain amount of time, and the period between these images is the same. As these images are developed, you can assess for systolic and diastolic motion, which basically means that you can see how the heart contracts and expands. Late gadolinium enhancement is another subtype of CMR, and this specifically allows for assessment of fibrosis. Late gadolinium enhancement, or LGE, is when a gadolinium contrast agent is inserted into the patient, and over a certain amount of time, it can detect if there's fibrosis. Fibrosis occurs when fibrosis can form when a patient has a heart attack, because this leads to, rather than them having normal muscle cells, it leads to them developing scar tissue, and LGE is the amount of scar tissue someone has. Over time, this LGE can increase, and this is negative for the patient as it can lead to adverse outcomes. There are many different factors to look for when a patient has HCM, one of those being LGE and the others being obstructions and aneurysms. The main point of my research is to look at how these factors can affect a patient with hypertrophic cardiomyopathy. The image here is an LGE of an HCM patient, and the arrows point to the areas where LGE exists. The white area shows that the person has fibrosis, and this is specifically in the mid-ventricular septum. This is significant because if the patient has it in their ventricular septum, then that could lead to worsening outcomes if they have HCM. Obstructions are also important to look for because this is when the muscles close in together and lead to a buildup of pressure. These obstructions can occur either in the left ventricular outflow tract, which is the area between the mitral valve and the left ventricle, or in the mid-ventricle. And to assess for if a patient has an obstruction, you can use echocardiography or CMR. Another type of obstruction includes that in the mid-ventricle, and this is when the obstruction is between the left and right ventricles. Any type of obstructions are predictors of negative arrhythmic effects. There are also apical aneurysms that are associated with hypertrophic cardiomyopathy. Aneurysms can occur anywhere, but in hypertrophic cardiomyopathy, it's usually in the apex. If a patient has an aneurysm, then they can also develop thrombus, which is a blood clot, and this can therefore lead to thromboembolic events, which can lead to a patient having a stroke. This is why aneurysms increase the risk of sudden cardiac death. Here is a CMR of an HCM patient with an aneurysm. As you can see here, this is the ventricular septum, this light gray area, and it's much thicker than it normally should be. This light gray area shows an aneurysm, and this is quite a large aneurysm. An aneurysm is when there's ballooning out of the muscle, and this CMR image clearly shows that. I've also looked at a few other methods, including computed tomography, or CT scans. These are used in assessment of hypertrophic cardiomyopathy less than CMR or echocardiography due to the fact that it leads to ionizing radiation, which is harmful to the patient, and it also uses an iodinated contrast agent, which can be harmful to the patient. This image shows a CT scan of an HCM patient. The arrow points to the part of the heart where there's a hypertrophy, and as you can see, it's in the ventricular septum that separates the left ventricle and the right ventricle. Machine learning is a newer type of imaging method in cardiology, and this uses artificial intelligence to characterize different parts of a disease. The aim of my study is to look at a few different factors, the main factor being which cardiac imaging modality is best for assessing HCM. As I mentioned before, there are many imaging methods that can be used for assessment of this disease, but I want to figure out which imaging modality is best for assessment of HCM. I also want to look at which imaging method is best for assessment of aneurysms and obstructions, as they may lead to worsening outcomes for a patient with HCM. And I also want to see how prognostic apical aneurysms and obstructions are for adverse risk assessment of HCM. By prognostic, I mean that if a patient has an aneurysm or an obstruction, how that could lead to the patient with HCM either having worse outcomes or better outcomes. I felt that the best way to carry this out was through a literature review, because this would allow for me to summarize which imaging method was best for HCM. To do this, I gathered many articles that specifically concerned HCM and many articles that looked at the different imaging modalities and compared which one was better. Most of these articles excluded patients with other cardiovascular diseases because if a patient has a disease such as coronary artery disease, then the sole reason why they have hypertrophy may not be due to the fact that they have a genetic mutation, but could rather be because they have another cardiovascular disease, which is why it was important for these articles to exclude all of the patients that did have those diseases. For my methodology, it also consisted of extracting many images that demonstrated either HCM, aneurysms, obstructions, and LGE, as aneurysms, obstructions, and LGE were the different factors that could lead to worsening outcomes of HCM. I also created basic models that show how all of these look in a patient. The first image here, the top image, shows a short axis CMR of a patient with a normal heart versus a heart with HCM. Short axis is an imaging method that allows you to look at the left ventricle, which is right here, and the right ventricle. And here is the ventricular septum. As you can see in this image, the ventricular septum is how it should normally be and is around 10 millimeters. But in the patient with hypertrophic cardiomyopathy, their ventricular septum has increased by a lot and is 23.6 millimeters. In a CMR image, usually the white area represents the blood pool and the dark gray area represents the cardiac muscle. The model that I created is shown down below, which shows the heart in a similar view, in the short axis view, and shows that the ventricular septum is normal here, but with a patient that has HCM, it's much thicker than it should be, and this can lead to worsening outcomes for the patient. Here I created a model of an obstruction, and an obstruction is present here when the muscles come in together, and this can lead to a buildup of pressure. This buildup of pressure can lead to an aneurysm forming, which is the ballooning out of the cardiac muscle. Usually the cardiac muscle would be here, but due to the aneurysm, which causes it to balloon outwards, it comes out like this. And the aneurysmal wall thickness is thinner than the regular wall thickness of the heart. Although you may believe that obstructions can lead to aneurysms due to the fact that it leads to a buildup of pressure, this isn't true. An obstruction doesn't always necessarily lead to an aneurysm, and an aneurysm doesn't always lead to an obstruction. Rather, the two are associated with each other. And in this image, the aneurysm was specifically in the apex. Here are more CMR images of a patient with hypertrophic cardiomyopathy. Images A through C show aneurysms that progressively get larger. This is shown by the light gray areas here, here, and here. As you can tell, the aneurysms are getting much larger. Images D through F use LGE to determine if the patient has fibrosis. Here in D, the fibrosis is quite prominent, as the white areas in the midventricular septum show that the patient has fibrosis. Image F is quite significant as well because this shows a thrombus at the aneurysm, which is seen by this arrow. The black spot shows a thrombus, which is also known as a blood clot, which can then lead to worsening outcomes for the patient. Here's a video of an echocardiogram of a patient with obstructive hypertrophic cardiomyopathy. As you can see here, this is the midventricular septum, and in HCM, it's a lot thicker than it should be. If this was a normal heart, then it would be a lot thinner, but here in the echocardiogram, it's much thicker. Over here, you can see these two leaflets closing in. This is a mitral leaflet, and here you can see systolic anterior motion. Systolic anterior motion is when the mitral valve goes toward the left ventricular outflow tract, and this is the opposite of what it should be doing. If the patient has systolic anterior motion, then they can have a worse obstruction, which therefore leads to worsening outcomes for the patient. Here's another echocardiogram of a patient with a left ventricular aneurysm that's presented by the white arrow. This is quite a large aneurysm, which is why you're able to see it well. But if it was much smaller, you may not have been able to see it well because although echocardiograms have great temporal resolution, they don't have as great of a spatial resolution as CMR or CT scans. Here, CMR and echocardiograms are being compared, and both of these images show a patient with HCM. In the top image, you can see a patient with HCM that has midventricular HCM with an aneurysm. This is an echocardiogram, and the midventricle is seen right here, and it's much thicker than it should be. And here is where the aneurysm is present. This is the same person and the same image, but rather it's taken through CMR. Here is the hypertrophied septum, and here's the large aneurysm. As you can see, due to the fact that CMR has greater spatial resolution, it's easier to identify the ventricular septum as well as the aneurysm. Here you can see concentric HCM. The first image is again an echocardiogram, and it shows a patient with hypertrophic cardiomyopathy in the short axis view. And although you may not be able to see it well, this shows how the muscle is hypertrophied or how the muscle has thickened. Here's the same patient, but this image is taken through CMR. Similar to the image above, it's easier for you to identify where the hypertrophy is due to the high spatial resolution. Color Doppler echocardiography is a type of echocardiography method that allows for a quantification of blood flow. This allows for a pressure gradient to be determined, and this is quite significant because if a patient has a left ventricular outflow tract obstruction or any type of obstruction, then their pressure gradient is much higher than they should be. It's important to quantify these values because if a patient has a higher value, then that could mean that they have a worse obstruction. After gathering all of these articles, I concluded that CMR is superior for detection of apical aneurysms. Again, this is because high spatial resolution allows you to look at any sort of aneurysm, no matter what the size of the aneurysm is. Echocardiography is superior in assessment of left ventricular outflow tract gradient when compared to CMR. This is due to the fact that you can quantify the pressure gradient, so it allows you to determine if the obstruction is worse. So that is one advantage of CMR and one advantage of echocardiography, which shows why it's important to use multiple imaging modalities for assessment of hypertrophic cardiomyopathy rather than one. Although in certain cases it may not be necessary to use multiple imaging modalities, such as if you're trying to determine if a patient has an HCM and it's clearly identified by echocardiography, which is the starting point, it may not be necessary to use CMR or any further methods. CMR best serves in assessing for areas of hypertrophy. Similar to how CMR can best assess for aneurysms, it can also best assess for hypertrophy because as you can see in image B, you can identify the areas of hypertrophy a lot easier than if you had an echocardiogram. And LGE, which is exclusive to CMR, detects fibrosis. No other imaging method allows you to detect for LGE, and this is quite significant because, again, if a patient has fibrosis or scar tissue, then that could mean that they have worsening outcomes for HCM. Apical aneurysms and obstructions are good prognosticators of adverse outcomes. So again, if a patient has an aneurysm, as represented in this image, and the TH stands for a thrombus because you can clearly see that here. If a patient has an aneurysm or an obstruction, then that could lead to an increase in risk of dying due to sudden cardiac death. I didn't look as much into machine learning as I did echocardiography and CMR due to the fact that machine learning is still a newer method compared to these two, but machine learning algorithms are able to identify morphological features of HCM. For example, it can assess for fibrosis. In this image segmentation, this is a deep learning image, which is similar to machine learning, but requires more computational skills to determine if the patient has characteristics for a certain disease. The top few images show a radiologist and how they were able to separate the different parts of the heart to determine if the patient had LGE, whereas the bottom shows how a machine was able to do it. And as you can see, it was quite accurate. Although again, my research didn't specifically focus on this imaging method. In my research paper, I also discussed treatment methods because I felt it was important to understand that as my research focused on a cardiac disease. There are a few different treatment methods, which include medication. The most commonly used medication is mavacamten, and this is quite effective because it decreases contractility of the heart. So as I mentioned before, when there are increased interactions between myosin and actin, there's hypercontractility. The way mavacamten works is it reduces the amount of interactions, which therefore reduces the hypercontractility and then leads to decrease of hypertrophy. Alcohol septal ablation is the insertion of alcohol which causes a controlled heart attack. You may think that this would be negative for the patient, but due to the fact that this is controlled, it can actually help the patient and it results in decreased hypertrophy, which allows for the patient's heart to be healthier. A myectomy is an invasive surgery that directly removes the hypertrophy. In this case, a doctor would surgically remove the hypertrophy and it is the most permanent method, yet it is also a very successful method. Finally, echocardiography is advantageous when compared to CMR as it allows for quantification of obstructions. CMR is advantageous due to the fact that it allows for accurate assessment of hypertrophy and fibrosis as well as aneurysms. And both aneurysms and obstructions are associated with adverse outcomes for patients. So if a patient has any of these, then it could lead to worse outcomes for the patients. I'd like to thank everyone that has contributed to my science research experience and thank all of the teachers that have helped me throughout these past few years. It's been a great experience for me, and again, I would like to thank my mentor for everything that he's done. I'm truly grateful for having established a mentorship with him, and he's put a lot of time and effort into helping me throughout my project, and I'm very fortunate for that. Thank you so much for listening to my presentation.
Please welcome Senior Emily Noonan to present her work on humpback whale acoustics.
Hello, my name is Emily Noonan, and thank you all for coming tonight. My research topic over the last 4 years has been humpback whale acoustics, and so I will be refreshing your memory a bit on some prior stuff that was gone over in a previous presentation. My mentor is Julianne Wilder. She's an employee of NOAA, and she's a cetacean acoustician. A cetacean acoustician is somebody who analyzes whales and other types of cetaceans and the sounds that they make. She helped me really narrow down my research project to humpback whales and helped me find what I needed for my project. So this would not be possible without her. Thank you. She always helped me figure out what to do when I was stuck. My research question is which themes are present in each season of the Martha's Vineyard buoy deployments? And the two seasons are December 2021 to June 2022, and December 2022 to January 2023. The goal of my research is to see which themes are present in these two different seasons during the Martha's Vineyard buoy deployment, and is to see if these themes have been carried on throughout the seasons and if they have evolved throughout seasons too. The second season is a lot shorter because unfortunately the buoy that we were using kind of went to the bottom of the ocean and we were unable to get any more data from it. But this is the best season for Martha's Vineyard to specifically hear these sounds because it is the beginning of the breeding season and male whales typically sing these songs to attract mates, which is very interesting. This buoy that I was using is located in Martha's Vineyard, which is in Massachusetts, for those of you who don't know. The gray is the land, the black is the water, and the star is where the buoy is specifically located. These buoys are very special. They were able to, it's a moored surface buoy, which I will go into later, which is another type of passive acoustic monitoring, but they have a hydrophone attached to them, and they're able to listen to these songs that are done during these seasons throughout the year, and yeah. Some of the literature that was very, very helpful to my project are when, and why do Western North Atlantic humpback whales begin to sing, and acoustic properties of humpback whale songs. These both were very helpful with my paper because they explored the properties of humpback whale songs and also a variety of factors by which they evaluated them in their own projects, which helped me decide on how to work on mine and how to analyze it the best way. The data that I used for my project came from passive acoustic monitoring, which was went over briefly earlier, but passive acoustic monitoring is a type of monitoring basically where they attach a hydrophone to a number of different things, including D tags or like archival tags, gliders, slocum gliders, and moored surface buoys, as well as a variety of other things, in order to hear what is happening in specific places or with specific organisms while they use the sounds. The ones highlighted in red are the two that I used specifically for this project, where I got my data from. The slocum glider and the moored surface buoy both digitally uploaded their data to a server, and which pinged to an archive online, which is the Robots for Whales website. And basically, that website has all of the spectrograms that I used for my project and the data that I used. I used the Robots for Whales website for my data. We selected the Martha's Vineyard buoy deployment in order to have a lot of this data to work with, because it was a fairly long deployment on the Robots for Whales website. There are many boxes showing the date and also the kind of whale that was detected during those days. The gray boxes are the ones where we did not see any whales that day or did not detect any. The red boxes are the ones where whales were definitely detected, and the yellow boxes are ones where whales were possibly detected, in which case they could not determine which kind of whale it was, or if this really was a whale or if it was just background noise. Because each whale has a different kind of sound that they make, and it's hard to tell sometimes if it's so far away, the whale, if they actually were there or if it was just some sort of background noise. Each time a new theme popped up in the Robots for Whales spectrograms, I would classify it as a new theme or a theme I already had, and I classified themes as themes A through N, which is a lot. Basically, I would go through each of these spectrograms of time, about 15 minutes every 15 minutes per day, and I would look to see if there were any common themes. Themes are snippets of songs said over and over again, like the chorus of a song, and basically they are said over and over again, which it's a discernible pattern which you're able to tell the song that they are singing, and that it's a theme for a specific day, and it's often repeated by other whales because they like it and they want to sing it to their mates too. I got sound files and listened to the themes, and I could see the corresponding spectrograms because I did end up using the Raven program for part of my data analysis, and it was very helpful overall to my project. I used the Raven program to run sound through and see the corresponding spectrograms, which allowed me to make a connection between the spectrograms that I was seeing and the sounds that I could hear, and it really helped me to understand what the things that I was looking at actually would sound like in real life. Unfortunately, a lot of the data was very, very low frequency, so I wasn't very much able to get a good clip of that, but I do have a clip, and I hope it works, I think maybe in the next slides, that I can hopefully play for you all. And this Raven program was from Cornell Labs, which was cool, and it was very, very helpful because it helped me to imagine what the themes sound like. Okay, so we're gonna see if this works. And if it doesn't, then I'll just explain. Yeah, it won't work. Okay, so the frequency on the y-axis is basically, it's measured in this one as 0.2, but in other spectrograms that I have, it's measured as in hundreds. This is so low because, well, the frequency is so low that you could barely hear it, so when they had to get this ready so people could actually hear it, they had to amplify the sound by a lot. So you get a lot of these little like things. They're basically echoes of the same note, and it could be another whale, but it's very, very unlikely that it's another whale. These main parts, the ones with the highest color intensity, are the important bits, and they're basically the main note that the whale sang that day. And then these are some down sweeps here and here and here, and then, yeah. So basically this was a similar theme, but from a different deployment, I'm assuming, for my project. And on the x-axis is time, which shows that this was about 54 seconds, this clip. Anyway, I used a Google Slides to analyze my data, and basically every day that there was a theme, I would put 1 or 2, however many times that theme popped up in that day. You can't see it, but on the right side for you is the date, and then I put themes across the top so you could tell easily which theme was on which day. And then over here we have one that is 3 days. I mean, three examples of a theme in one day, but I also found up to 7 examples of a singular theme in one day, which means that the whales probably really liked that theme. This is a screenshot of the chart I used to keep track of my data and what themes were seen on which days, which was very helpful organizationally. There were a variety of themes shown between both seasons, and there were multiple common themes between both seasons. This is an example of one of the themes that I saw on the Robots for Whales website. It is theme G, that I classified as theme G. The frequency, once again, is, well, it's shown by 100s, and this shows that just above 200 hertz is the main song here. And some of these, like, really sticking up straight ones, those are not actually whale sounds, they are artifacts. Artifacts are basically a glitch in the system, and they are unimportant. They do not mean, I mean, there's probably some little notes here, but nothing, like, crazy. It's just very glitchy. But, yeah. So there are some down sweeps here, and some up sweeps we got here. They're very small because the whale was most likely very far away when we were recording this, and it did not, because as the whales get closer, you see the themes longer, like the notes individually, the notes. Okay. Here is yet another example of some themes I saw on the Robots for Whales website. So here we have a lot of the artifacts. So basically, we are just looking at these little up sweeps here and down sweeps, and here. It's hard to tell with some of these longer themes and some of them that have most of these artifacts, because you cannot really discernibly tell the difference too much between the artifacts and the actual sound that the whale made. Thankfully, I had other examples of theme L and was able to easily identify this, and this just seemed like one of the more interesting images that I had to show. But here's a down sweep, and you can tell that it's just an artifact going down. Now, this is a good example of themes that were shown to evolve between seasons. Both of these have the same down sweeps between both seasons. Here, they're 2 different seasons these spectrograms came from. But theme H and theme H2, and then sometimes theme H3, were all evolved versions of theme H. Basically, they all had these same underlying down sweeps, but they had some different up notes up here and here, which means that they were similar, but not exactly the same, but similar enough to be considered the same theme. And I have another example for you. This is theme D, which I also thought was very interesting, because it could be a duet or possibly an echo of a singular whale, here shown with these 3 up sweeps. And basically, this is a theme that I had in the yellow box. And this was repeated multiple times throughout the song, throughout that day. There were a lot of common themes between the seasons, and you can see where the evolutions between these seasons occurred in themes like Theme H and Theme H2, because they were very, very similar and they had the similar base, but they had some differences between them. These themes were evolved over time and changed where they were not unrecognizable. Turnover rate, which is basically the amount of time it takes something to completely change, was not one season because we saw evolution, but we did not see complete 180. We did not see different themes between seasons. And in the future, I will be attending college at one of these two places, Roger Williams University or University of Maine, and I will be majoring in marine biology with hopes to continue my research. And before I finish, just a big thank you to my science research teacher for the first 2 years, Dr. Forsberg, and the most recent one, Ms. Lavazoli, and then also a big thank you to my mentor, who this project wouldn't be even happening without her. So thank you so much. She has dedicated so much time. Thank you.
Our last presentation of the evening will be from Senior Emily Robinson, the valedictorian of Class of 2024. She will be speaking tonight about her research on socioeconomic status and cognition by race.
Hello, my name's Emily Robinson, and tonight I'll be talking to you about my research paper. Here, this image shows the brain of a person with dementia, as seen on the left, compared to the brain of a person without dementia, as seen on the right. As you can see, the brain of a dementia patient is considerably smaller than the brain of a healthy individual. In this presentation, I will be discussing how different socioeconomic factors lead to ethnic disparities, specifically between non-Hispanic Black people and non-Hispanic white people, for the cognitive decline that can be seen in this image. My mentor is Dr. Indira Turney, who is a postdoctoral neuroscientist at Columbia University Medical Center. She's part of the TAB Institute for Research on Alzheimer's Disease and the Aging Brain, and she mainly studies how life experiences contribute to brain health disparities among racially diverse middle and older adults. Dr. Turney has been extremely helpful with my research. Beginning in my junior year, she began to meet with me over Zoom about once a month to help me dissect peer-reviewed articles and give me advice on my presentations. Going into my senior year, she's been even more helpful by providing me with the resources I've needed to write my research paper. She's also helped me keep an organized timeline of the goals I had hoped to accomplish and has answered any questions I've had in the writing process. One of the most critical problems addressed by medical sociologists is that society's less privileged members live in worse health than the more fortunate ones. This is due to differences in socioeconomic status, or SES, which is determined by economic and social factors such as education, income, occupation, place of residence, and ethnic origin or religious background. Socioeconomic conditions across all stages of the life course, from as early as childhood, appear to make unique contributions to cognitive function in midlife. This suggests that disadvantaged socioeconomic circumstances faced during childhood may be overcome to a certain extent by upward mobility later in life. Recently, variations in cognition among diverse populations have gathered a lot of attention. Research has shown that older Black individuals have a potentially faster rate of cognitive decline compared to older white individuals. This is due to the fact that older Black individuals have a higher prevalence of SES-related risk factors for cognitive impairment, such as lower education, less employment opportunities, and more unstable communities. While many studies on the impact of SES on cognition have focused on education, many of these studies have overlooked other SES factors and have looked at late life rather than midlife. Given the lack of knowledge on how distinct SES risk factors work together to explain differences in cognition among contrasting races in midlife, this study sets out to address this gap in knowledge. After meeting with my mentor in the beginning of December and going over the necessary variables that I needed to look at, I was able to readjust and develop a final research question, which is how does current income and educational attainment influence memory performance among middle-aged Black and white adults? This paper included participants who were a part of a community-based study of cognitive aging that enrolled middle-aged adults from the Offspring study of racial and ethnic disparities in Alzheimer's disease. Previously, full reports have been reported, but however, in short, those who were enrolled in the Offspring study were dementia-free at baseline, age 30 or older, were fluent in English and or Spanish, and were either African American, white, or Latinx. Before starting an analysis, the original data was modified to fit the study. The specific variables focused on in the study were cognition, which was memory, education, which was less than high school, some sort of college or postgraduate, and current income using $35,000, which is the median individual income in New York City reported in the 2020 Census Bureau. Then, after deciding coming up with my variables, I figured out the covariates that I wanted to control for. A covariate is an independent variable that can influence the outcome of a given statistical trial, but which is not of direct interest. For this study, the covariates that were controlled for were age, sex, which was male versus female, and health status, which included diabetes, hypertension, clinical stroke, or heart disease. Since the establishment of the Offspring study, over 1,500 participants have enrolled. However, for this analysis, after filtering out certain characteristics, such as excluding Latinx participants, the sample size was reduced to 638 participants. Then, after filtering for the variables and covariates, the sample size was further reduced to 565 participants, with 326 being non-Hispanic Black and 239 being non-Hispanic white. Here, this table shows the breakdown of the number of people in each category of income across and within race. Within the race, you can see that 42.9% of non-Hispanic Black people had a lower income, while 57.1% were higher income. In comparison, for non-Hispanic white people, only 14.4% were lower income, while 85.6% were a higher income, showing that non-Hispanic white people were more likely to have a higher income than non-Hispanic Black people. This table shows the breakdown of the number of people in each category of education across and within race. Here you can see for non-Hispanic Black people, 32%, or about a third, had a less than high school education, while 51.2% had some sort of college education, and 16.8% had a postgraduate education. On the other hand, only 3.4% of non-Hispanic white people had a less than high school education, while 42.5% had a college education, and 54.1%, or more than half, had a postgraduate education. This shows that non-Hispanic Black participants were more likely to have a less than high school education, while non-Hispanic white participants were more likely to have a postgraduate education. The significance that age and sex had on memory scores across non-Hispanic Black participants for income is shown in this table. The p-value is used to measure significance, and a p-value less than 0.05 usually indicates that something is statistically significant. As you can see, age has a less than 0.001 significance, meaning that age was very significant, and as participants got older, memory scores worsened. You can also see that income was statistically significant, meaning that lower income led to lower memory scores. Also, trending-wise, overall, men had worse memory scores than women, and when controlling for chronic health conditions, there was no significant difference, which is why it's not included in these tables. For non-Hispanic whites, the significance of age was less than 0.001, showing that, again, age was statistically significant. However, for income, there was only a 0.105 significance, showing that income did impact memory for non-Hispanic white participants, but not as much as it impacted non-Hispanic black participants. Again, trending-wise, although the significance was less than non-Hispanic black people, men overall had worse memory scores than women. The significance that age and sex had on memory scores across non-Hispanic black participants for education is shown in this table. As you can see, again, age was statistically significant, and having some sort of college education resulted in higher memory scores, along with having a postgraduate education. Again, trending-wise, men overall had worse memory scores than women. For non-Hispanic whites, the statistical significance of age, again, was less than 0.001, showing that as participants got older, memory scores worsened. However, when you look at the significance of education, you see that it didn't have a big impact on memory scores for non-Hispanic white participants, with a significance of 0.255 and 0.165, showing that education had a much greater effect on non-Hispanic black participants than non-Hispanic white participants. And again, trending-wise, overall men had worse memory scores than women. The disproportionate effect of SES factors on memory may be explained by the substantial racial disparities in opportunities and resource attainment experienced in history. One possible explanation for this is the resource substitution hypothesis, which states that education compensates for decreased access to other resources, such as power and earnings available to advantaged groups. This theory implies that racial minorities, due to structural disadvantages, would benefit more from increased education than non-Hispanic white benefit. The results also indicated that age had a statistical significant effect on memory for both non-Hispanic black and non-Hispanic white participants, though the effect of age was even greater on non-Hispanic black participants, which may be due to accelerated aging. Discrimination and less economic security for black people can lead to higher levels of both physical and psychological stress, which can cause a quicker aging, which might explain this result. In the fall of this year, I will be attending Stony Brook University, which has honestly been one of my top choices since the beginning of middle school. I will be majoring in physics, where I've been welcomed into the Department of Physics and Astronomy in the College of Arts and Sciences, and I'll be a part of the Women in Science and Engineering, or WISE program, where I will be able to work in a small community that will allow me to work closely with peers and form close friendships, and I will also be able to work with mentors and continue my research journey. Before I wrap up, first I'd like to thank my mentor, Dr. Turney, who's been extremely helpful. I would not be able to get this far in my research without her. I'd also like to thank Ms. Vicini for helping to put the whole symposium together, and I'd like to thank all of my science research teachers who have helped me in my research journey in the past 4 years. So thank you to Dr. Forsberg, Ms. Lavazzoli, and Dr. Z for stepping up in their place. That concludes my presentation. Thank you for listening.
I was right? More hopeful, more inspired, your head hurts from all of the things you learned tonight. Let's have a round of applause for all of our science research presenters tonight. What I'd like to do at this point is invite our juniors and seniors to the stage for a brief question and answer period. You're going to have the opportunity, ladies and gentlemen, to ask a question. Can we bring the house lights up a little bit, please? And my underclassmen hosts, who gets the microphones? Okay, juniors and seniors, come on up. What we'll do is open the floor to questions and answers. After that, Mr. Richard is going to offer his thanks and remarks as our students are out at their posters.
No, I was like,
Test this.
someone's mic isn't on.
Okay. Yes.
Mr. Clemenson.
Who's got a question? Don't hold back.
I have a question for Emily Robinson. Yeah. How did you take into account the biracial people? Were they under like the non-Hispanic black people or the non-Hispanic white people?
Ask that again?
What category did biracial people fall under?
Well, it was like self-reported, so I guess they would choose which race that they thought they would fall under. But honestly, I excluded a lot of the participants because I like narrowed down the participants that I included. So there was over like 1,500 participants in the original study, but there was like a lot of factors that I took into account because I narrowed it down to only 565 participants. So they were self-reported, so.
All right, thanks.
Hi, this question is for Chris Roldan. Can you give an example of how acoustic data collection can help save the whales?
Okay, so data collection is very useful. As I presented in my presentation a bit earlier, it's one way that we can use acoustic information for conservation efforts is knowing where we would be able to, y'all can hear me, right? Okay. It's how we are able to direct efforts towards conservation. Like, for example, in the, I believe it was blue whales that were fitted in the middlemost region in the areas of New York or New Jersey. If it comes down to if the blue whales are endangered or in need of any assistance, we will be able to know where they are because that acoustic information that we have gathered shows us that they are going to be there at this specific point in time in the year. Thank you for the question.
Thank you. Hi, my question is for Cassidy. What other adverse events or side effects may be caused by fingolimod?
Yeah, so can you guys hear me? Other adverse events may include increased risk of infection, which as mentioned was due to the decrease in lymphocytes, and then other adverse events may include a slowed heartbeat or bradycardia, and this is caused by receptors on cardiac myocytes being targeted, and these myocytes control contraction of the heart, and so when these receptors are modulated, it no longer can function to the full capacity and allow for a proper heart rate. And then other adverse events could also include back pain, abdominal pain, or weakness.
I have a question for Dylan Foley. How beneficial was it to your research to have hands-on experience with your horseshoe crabs?
It was actually very beneficial because it's like getting the experience in the field is not like reading you really understand the information being thrown at you more when you're in the field. Because it's more the reality of the situation. You're dealing with the horseshoe crabs instead of reading about them, which is, in my opinion, pretty cool. You really get to link the information you read about and kind of understand it more in the field because you kind of grasp this situation more, I guess. But thank you.
You guys all did such a great job tonight. Congratulations, and especially on your amazing futures. I have a question. What would you recommend to the younger classmen that are following behind you, having had this experience for the past couple years?
Have fun. It's a research class. Have fun. Choose your own topic that you're interested in. Have fun with it.
I would say going back to the question recently asked to me, I would say get involved in field research, because if you have that opportunity, I know many people do bibliographic studies and present based on that, but if you really get the hands-on learning, you build that understanding and you will fall in love with your topic.
Also, you have to pick something that you're really passionate about doing and something you actually care about, because if you choose something you don't really care about, then you won't love what you do or find it so interesting. But we all chose something that we really enjoy at least learning about, and it really makes a big impact on our futures, I think.
Hi, my question's for Emma. So I had a question on how you decided to track turtles instead of other migratory species, and how turtles are more indicative to a bigger climate question in terms of migration and also just ocean health and the creatures that live in it.
Thank you for your question. When I picked my topic sophomore year, I tried to get a lot of hands-on research, so I decided to participate with the New York Marine Rescue Center, and there they kind of gave me like a baseline with everything on all my research, and they just showed how like sea turtles specifically are most like detrimental to like weather events. They also have like other animals there like dolphins and stuff like that, but they mainly focus on sea turtles, and that kind of like paved the way for me. So.
Don't worry, Dylan, this isn't for you. So just wanted to echo and say, you guys are all amazing. Congratulations. Just mind-blowing. So great job, everyone. This question is for Isabel. So, you know, obviously we live in a beach community, and we see the piping plover nests all over the place. Obviously, one of the things we can do is keep our dogs on the leash. What else could we as a community do to help the plovers?
So that's a really good question. The plovers are a very fragile bird, and just the smallest bit of disturbances can affect them in such a great way. Even things during the winter, such as driving on beaches, can have such a big impact on them when they come back, because it just destroys the dunes that they live on, and all the substrates that are in the sand are so important for them. And while driving on beaches, it really just like compacts that sand a lot. It makes it so much harder for them to find good areas to nest in as they come back. So driving on beaches is something that you should try to avoid for the most part, as well as making sure to stay out of the dunes while walking on the beach or any other sort of beach activities that you're doing.
Ladies and gentlemen, thank you for your questions. Juniors and seniors, let's have a round of applause for their well-prepared answers and research. I'm going to invite all of our researchers at this point to exit the stage and those in the audience to head to your posters. So you can do that now and ready yourselves. We will invite in just a few moments all of you to wander through the poster gallery. The hallway and the cafeteria are transformed, and there's some dessert and refreshments there. But before we do that, and as the students get themselves ready, I'd like to introduce to the microphone our high school principal, Mr. Richard.
Okay, good evening, ladies and gentlemen. I'm going to keep my comments brief as we're heading towards my favorite portion of this evening, which is the poster discussions. So I just want to talk thematically very quickly about what's happened here. 11 years ago when this program began, I talked about this being the realization of a professional dream for me. My passion is education and learning. And you saw students with their passions in all their respective fields of study. And while they're excited about all their various fields of study, whether it be dealing with cures or conservation or efficiencies in the world, right, I look at this as an incredible learning experience, a journey that began 4 years ago that resulted in 12 college credits, graduate level research, and much more. The alumni of this program are currently engaged in graduate work. They're in medical school. They're at Ivy League colleges doing some really amazing things. And I'm looking forward to hearing what all of our graduating seniors from our research program are going to do out in their collegiate studies and beyond. So at this time, what I'd like to do is welcome everyone to join us in the hallway and the atrium over here and the cafeteria where we have some dessert set up. The best part of the evening is talking to each of the individual students about their research in its various stages of development. So I encourage you to meet with as many of our students as you can and have a great night. Thank you for joining us. Take care.